So I'd like to introduce John Abelson, who's going to chair this morning's session. John figured out one of the very first RNA splicing mechanisms. It was at the same time we were figuring out how tetrahymine or ribosomal RNA underwent splicing. John and his group in Southern California, figured out how yeast tRNA splicing occurred. And he was less lucky than we were because it turned out that it was traditional protein endonuclease and ligase that were responsible rather than RNA catalysis, but it could have easily been the other way around. And John went on to use the awesome power of yeast genetics, he and his wife Christine Guthrie, and really dissected a lot of the features of messenger RNA splicing using this genetically tractable system. John is also known for having discovered the spliceosome, the large complex whose structure we're going to hear about this morning. So great to see you, John, and he's going to introduce Ansia Park, who is the first speaker.
Yeah, well, I'm sorry that Joan is not here because she was also there at the very beginning of splicing, and perhaps because it was a lot of nice people that we made a really good field, I think, and it's continued to be successful. The speakers today are going to talk a lot about we finally got the structure of the spliceosome, and it was because cryo-EM really came of its own. And so our first speaker is Ansia One, right? And she will tell us the really very interesting things that she's been doing in Joan Steitz's lab.
Hi, thank you for the introduction and for the organizers for the opportunity to present at this wonderful meeting. My name is Ansia Park. I'm an MD PhD student co-mentored by Joan Steitz and Akiko Iwasaki at Yale. And because I know how much Joan was looking forward to this meeting and sharing our latest update on a class of non-coding RNA called dog RNAs, I will do my very best to deliver the story that she was going to present today. So the work that we wanted to share is about our findings of elevated dog RNA expression in neurons that revealed their possible role in regulating retrotransposon transcripts. So although only 1.5% of the human genome codes for proteins, we now know that greater than 70% of the genome is pervasively transcribed, leading to generations of thousands of non-coding RNAs. What is remarkable about non-coding RNAs has been that discovery of each new type of non-coding RNA has challenged our previous understanding of RNA biology and revealed completely new functions. And I know that I do not need to highlight this fact to the community here, but reading this very special review by Joan and Professor Check, this was what got me personally excited and motivated to study a new class of non-coding RNAs that Joan and colleagues had discovered in 2015 called dog RNAs. So what are dog RNAs? Dog RNAs are produced by read-through transcription that does not terminate but continues beyond the gene ends for thousands of kilobases. In various stress conditions, read-through transcription dramatically increases for a subset of genes, leading to generations of thousands of long non-coding transcripts that are continuous with the upstream gene body. We have collectively called these dog RNAs because they contain the downstream of gene sequences. Unlike mRNAs, dog RNAs remain in the nucleus and are thus not translated. Other than that, however, their function or their impact of expression is not known. Dog RNAs have now been described in a variety of different stress conditions, including heat shock, osmotic stress, viral infections, and cancers. I should note here that not all genes, but about 10 to 20% of genes are read through, and from each gene, only a fraction of transcripts are read through. However, what is remarkable is that dog RNA as a group account for almost up to 30% of all intergenic transcripts, which suggests that dog RNAs likely represent a major new class of long non-coding RNAs. So during my PhD, I set out to understand these curious collection of non-coding RNAs with the goal of answering these three questions: their expression profile, their characteristics, and their function in human tissues. First, dog RNAs have thus far only been studied in cultured cells, and it is not known whether they have regulated expression in human tissues. So to address this, we systematically investigated read-through levels across human tissues using the GTEx dataset by examining the number of cellular genes that are read through at steady state for at least 5 kilobase. And these genes are called read-through genes, such as an example shown here, which has reads beyond the gene ends in certain tissues producing dog RNAs. Comparing the number of read-through genes across 49 different human tissues using the GTEx dataset, we saw that different tissues showed varying levels of read-through transcription at steady state. Notably, it was most prevalent in the cerebellum, which had the greatest number of read-through genes. When we normalize this number to the number of expressed genes in each tissue, read-through genes uniquely represented a high percent of expressed genes, of about 10% in the cerebellum. Analysis of mouse tissue revealed a similar trend. We saw that cerebellum had higher number of read-through genes at steady state compared to other adult tissues. And using mouse tissues, we also saw that at the individual gene level, a significantly higher fraction of transcripts were read through in the cerebellum compared to other mouse tissues. So this indicates that dog RNAs are most abundant in the cerebellum, not only in terms of the number of dog RNA species, but also the abundance of individual dog RNAs. We next looked at dog expression across cell types within the cerebellum and saw that neurons, in particular the granule neurons, had the highest read-through levels at steady state, and that majority of dog RNAs in the cerebellum were produced from the neurons. Before we went on to explore this further, we wanted to ensure that the elevated read-through levels in the neurons indeed represented true expression of dog RNAs. So we examined these transcripts for features that define dog RNAs. First, dog RNAs are by definition continuous with the upstream gene body and nuclear. Second, neurons are known to produce transcripts with extended 3' UTR, so we wanted to distinguish these products from the non-coding dog RNAs. And lastly, dog RNAs are known to respond to stress. So first, by performing FISH with probes that span the entire region of dog RNA, we confirmed robust expression of dog expression in neurons in the cerebellum and saw that these transcripts predominantly localized to the nucleus of neurons as expected. We also confirmed biochemically that dog RNAs are much more nuclear than their mRNA counterparts by isolating granule neurons and sequencing the fractionated RNA. Because neurons, as I mentioned, is known to produce transcripts with extended 3' UTR, we directly compared the lengths of dog RNA to those of 3' UTR isoforms that were experimentally identified by binding of cytoplasmic poly-A binding protein in cerebellar neurons. And we saw that dog RNAs are not only nuclear, but also significantly longer than the 3' UTR isoforms, indicating that dogs are RNA molecules distinct from the neuron-specific 3' UTR isoforms. And additionally, using long-read sequencing, we directly observed long reads that span from the gene body and continue and extend into the downstream of region in the nucleus, validating the presence of bona fide dog RNAs in cerebellar neurons. Lastly, we asked whether read-through transcripts constitutively expressed in the cerebellum further respond to stress or conditions known to induce dogs in cell. So to this end, we modeled hyperosmotic stress in mice, which was the first condition dogs were discovered in, by injecting hypertonic sodium chloride solution and causing hypernatremia or increase in blood sodium levels. Steady state read-through levels, which was highest in the cerebellum, as I previously mentioned, further increased in response to hypernatremia. And furthermore, genome-wide sequencing showed the majority of dog RNAs that are constitutively expressed in the cerebellum are upregulated in response to hyperosmotic stress, whereas the levels of the mRNAs were largely unaffected, indicating a selective increase in transcriptional read-through. So the response of these transcripts in cerebellum to canonical dog-inducing condition further argues that these transcripts are bona fide dog RNAs sensitive to physiologic stresses. So to summarize the first part of the talk, we established that dog RNAs have versatile expression in different tissues, cell types, and contexts, in that they are not only stress-inducible, but also constitutively expressed at elevated levels in cerebellum and in neurons in particular.
So I'd like to introduce Reinhard Luhrmann, really old friend of mine and with many of you. Reinhard and I have drank and danced all over the world at various attractive meetings, and we've also struggled with trying to understand the early features of the spliceosome and finally its structure. And we now have a number of structures. I think they are both fascinating and baffling at the same time. So there's, we don't understand everything, but I bet we understand much more after we hear Reinhard's talk.
And I would also like to thank the organizers for inviting me to this wonderful meeting. It's a real honor to be here. And it's also a great pleasure to see old friends of the RNP community here on this occasion. So the first slide is just to remind you that nuclear pre-mRNA splicing involves just two simple phosphoester transfer reaction and proceeds via two intermediates after the first catalytic step, namely the excised exon 1 and the exon 2 intron lariat with the branch structure here, and then follows the mRNA product formation. So it has the same chemical pathway as group 2 self-splicing introns. However, for pre-messenger RNA splicing, we need myriads of factors that form the spliceosome, and one particular important family of factors are these snRNPs, U1, U2, U5, which contain only one snRNA each, and then the U4, U6 diSNRP, in which the U4, U6 RNAs are base paired together. Under splicing conditions, the latter two particles form a pre-assembled U4, U6, U5 tri-snRNP complex, and these color boxes show all the proteins associated specifically with one or the other snRNP. Now, the spliceosome has one striking feature, and that is that it doesn't exist as a pre-assembled enzyme in the cell. Instead, all these factors in snRNPs and additional splicing factors have to be recruited anew onto each pre-messenger RNA substrate that needs to be spliced. And this is summarized schematically in this splicing cycle here, where I focus on the fate of the snRNPs and a set of RNA helicases which use ATPase to give directionality to the splicing reaction. So we can distinguish roughly 4 phases. Initially, the snRNPs are recruited stepwise to the intron, and this cumulates here in the B complex, which doesn't have an active site yet. So this is the other major feature of the spliceosome. You have to create an active site each time during each round of pre-messenger RNA splicing, because none of the building blocks of the spliceosome has a preformed active site. And this happens during the activation phase here, whereby BER2 RNA helicase kicks out the U4 RNP, and U6 now remodels to form a catalytically active molecule chaperoned by U2 RNA. And then following remodeling, we now enter the catalytic phase with the 2 catalytic steps happening here and ending in the P complex from which the mRNP is then displaced, and the resulting intron lariat spliceosome is now dissociated, delivering the parts to a fresh round of splicing. When in the past we started purifying and identifying these various assembly stages here and did the proteomics, it turned out that it is a very protein-rich molecular machine with more than 170 proteins around, but was more interesting even is that there is a constant fluid of proteins hopping on and off the spliceosome during the transitions from one major step to the next complex. And this reveals that the spliceosome is a very dynamic and fluidic structure. Yet in the last couple of years, it has been possible to reconstruct high-resolution cryo-EM structures of most of these major assembly intermediates, both from human and yeast, and these were primarily reconstructed in the labs of Jigong Shi in Beijing, in Kiyoshi Nagai's lab in Cambridge, and in Göttingen by us in collaboration with Holger Stark. Now, the structures, together with the results from decades of biochemical and genetic studies, have provided us now with an unprecedented insight into the mechanism of pre-messenger RNA splicing. However, this is not at all the end of structural biochemistry, because as I indicated, there are lots of proteins being exchanged between these transitions between the major assembly intermediates here. Sometimes these can be tens of proteins, and therefore in the future we have to further dissect now these intermediate stages in order to understand fully the trajectories of all the components of the spliceosome and how they are regulated in these exchanges. Now, when Tom Check wrote to me I should drop about 30 slides from my initial collection, I then I decided I skip the entire second part of this cycle and focus just on the initial assembly pathway up to the activated spliceosome and give you a quick tour through how the spliceosome is assembled. And in the second part, I will then particularly discuss the transition between the B and B-ACT, because here we could identify and isolate now intermediate assembly stages, and which gave us insight for the first time into how a U2, U6 catalytic structure is being assembled and how proteins appear to chaperone the final conformational state of the active site. Okay, so let's start with the early stage, and here U1 and U2 join each other and enter this pre-messenger RNA intron and get stably bound here at the level of the so-called A complex. U1 base pairs to the 5' splice site, and U2 now base pairs to the branch site, and thereby the branch site adenosine is bulged out, and this extended branch site U2 RNA helix is now stabilized by several proteins of U2 snRNP, and the most important one is this SF3B, which has a superhelical heat domain, and via its terminal heat repeats, it clamps down on the extended part of this U2 branch site helix. And what you can see here is that the bulged branch site adenosine is now tucked away here in a pocket of amino acid side chains. And this kind of chaperoning away of reactive sites of pre-messenger RNA as well as snRNAs is a general principle which we encounter in the spliceosome. It's a very careful machine, and these reactive sites were made available just on time when these sites are needed for function, and often this happens due to the helicase action or sometimes post-translational modification. And this grip of the SF3B and other proteins on this branch site U2 helix will be kept throughout the entire following assembly of the spliceosome up to the catalytically activation of the spliceosome. And we will often see this 5' domain in the next couple of structures. So the next particle which joins now the spliceosome is the trisnerp, and this is the cryo-EM structure, and the trisnerp is obviously the largest pre-assembled complex, and it contributes also several components to the active site of the activated spliceosome, including the U6 RNA, but as you can see here, U6 is heavily base paired to U4 RNA, and U4 is a kind of chaperone which helps to prevent U6 RNA to prematurely be activated. Moreover, as I mentioned, BER2 RNA has to dissociate this at the onset of activation, but in the cryo-EM structure, the BER2 helicase is located outside here, and the substrate for the BER2, the U4 RNA, is located here about 10 nanometers away. That means, again, in the trisnerp, the proteins chaperone the RNA for the time being into a silent, functionally silent conformational states, and this will have to be opened by remodeling events. We showed previously, what you see here is a major player of the spliceosome and also of the trisnerp. This is the PRP8 protein, and this has 4 domains which we have to learn now because they are important now during the activation. It has an amino terminal domain connected to this so-called large domain, which contains an endonuclease and reverse transcriptase-like domain. And then we have 2 smaller domains, the RNase H-like domain. This is a hub of protein-protein interaction and a very mobile tool of PRP8 to stabilize emerging RNP structures. And then the last small domain is the JAB1-MPN domain, which binds tightly to BER2 and helps to keep BER2 on the spliceosome during its large-scale translocational movements as the spliceosome assembly proceeds. Now, the orientation of the large domain towards the anti-D domain shown here is as in the trisnerp, and we call this the wide open conformation. We refer to this cavity here. This is an important site where the active site will be accommodated finally. In the B complex, the large domain has moved towards the anti-D domain, and we call this the open conformation. And finally, in the mature BAC complex, we have a restricted opening here, the cavity, and this is the closed conformational state. And this dynamics of the large domain plays a major role in the formation of the U2, U6 catalytic site, as I will discuss later. We showed previously that the initial encounter of the trisnerp with the A complex, or vice versa, is a loose one and happens only at the RNA level. We form here a U2, U6 helix between the 3' end of U6 and the 5' end of U2 RNA. And the decisive step now to form the stable B complex is the PRP28 mediated dissociation of U1 RNA, base pairing from the 5' splice site, and instead a sequence ACAGA, the ACAGA box, which is located upstream of stem 1 of the U4-U6. This is now base paired again to the 5' splice site. And this is now the stable situation where for the first time we have committed now a particular branch site with a particular 5' splice site for interaction during the activation. What we also showed that during this step, a set of roughly 10 proteins now are stably integrated, and I will discuss the role of these 4 which center around PRP38, as we learned an additional role only recently during this activation process. And when we had the first cryo-EM structure of the trisnerp, as well of the B complex, it became clear that most of the components of the trisnerp undergo significant remodeling, and some of them are shown here. Here we have the BER2 RNA helicase positioned close to the RT domain of the large domain, and this will move over by about 20 nanometers now, and at the same time the large domain moves towards the anti-D domain, now providing binding sites for this set of 4 B-specific proteins which bind into the interface between the large and the anti-D domain. And if you had time to watch this guy here, it has turned around by about 180 degrees and has now adopted a completely new protein-protein interaction situation. This is now what comes out of these remodeling events. This is the B-complex cryo-EM structure, and what has happened now, the BER2 has moved over, and during this stage it has now accommodated the single-stranded U4 RNA upstream of the U4, U6 helix here. However, at the same time, we have recruited the FBP21 B-specific protein, which docks onto the ACAGA 5' splice site helix. And this is a kind of roadblock. This first has to be pulled away to allow now BER2 to move into the helix here. U2 has been recruited here, and it is docked onto the remodeled tri-SNRNP. And please note that the 5' part of U2 with the SF3B heat domain and the U2 branch site structure is still far away from the center of PRP8, where the U2, U6 helical catalytic site has to be created. So we have to move down the U2 quite a distance to this region. And what we observe here is the position now of these 4 B-specific proteins. And you can see that the ACAGA 5' splice site, in addition to the extended part of this helix, these are docked now here in the B-complex on top of the amino terminal domain. And these 4 proteins, on the one hand, stabilize the open conformation of PRP8 and at the same time stabilize the extended part of this 5' splice site helix. And this is really essential, as we could recently show. If this is shortened or we don't have these proteins, you will not activate the spliceosome. Okay. Now we are close to the activation, and before I discuss now the stepwise formation of the catalytic site, I think it is necessary that I first show you the end product, so the mature B-ACT complex, how this looks like, and how the U2, U6 RNA is accommodated in the activated spliceosome. And so this is started by the BER2 RNA helicase. It dissociates U4 from U6 RNA, and U6 RNA now remodels. It forms the U6 ISL, bulged out, a uridine residue. And on the other hand, it can now interact with U2 RNA to form this U2, U6 helix 1, which we dissect in helix 1A and 1B, and the 1B contains the AGC triade in the U6 RNA, and this receives 3 Hookstein interactions with 3 bases by Hookstein interactions to form a so-called catalytic triplex, which then binds the 2 magnesium ions and which catalyze. And this has finally nailed down about 10 years ago by Judy Piccirilli and John Staley using RNA biology techniques, demonstrating finally that indeed the spliceosome is an RNA catalytic enzyme. Okay, and what we showed that during this stage from B to B-ACT, about 50 proteins are exchanged. That means, on the one hand, all the B-specific proteins which we have just taken on board are now displaced, are no longer present. All the U4, U6 proteins are dissociated, as well as about 5 U5 protein. On the other hand, we now take 25 new proteins on board, including these complexes of the PRP19 or NTC complex, NTC-related proteins, the RAISE complex, and the IBC, the intron binding complex with the Aquarius helicase, and then some additional late-acting B-ACT factors. And with no surprise, the resulting B-ACT structure is now, of course, a much bigger entity and has little similarity with the starting complex of the B type. I don't have the time, fortunately, for you to go into the details of this structure, but what has happened here is that now we have created in the B-ACT complex for the first time a catalytically active U6, U2 RNA structure. And let me see whether I can get this activated. Apparently not. Well, then we have to discuss it. So down here is the ISL structure, and these in magenta are these three nucleotides, the so-called catalytic triad, and this forms with the U2 RNA the helix 1b, and here this is continued by the helix 1a, and please note that the nucleotides of the helix 1a in U6 RNA are located here in a sharp turn of U6 RNA, the U61A loop, and I will come back to this because this is the last event. This is the last event during the final maturation of the U2, U6 RNA remodeling to get a mature BACT complex. So these sophisticated structure will not form or be stabilized without proteins, and the major protein important for this is PRP8, which now has adopted the closed structure with this restricted cavity here. And the RNA, the U2, U6 RNA network is now docked into this cavity as shown here. And in the next two panels, I'd like to show you two important points. So this sophisticated structure, namely, docks now to two distinct sites of PRP8. On the one hand, the ISL structure down here is docked to the bottom of the NTD domain. Here we have the U6 Akara box helix on the top of the NTD domain. And the U2, U6 helix 1 is now docked on the large domain at the helical bundle and RT domains. So these two sites, secondary structural elements, are docked to distinct sites of PRP8. Okay, and then of course the newly recruited proteins such as CDC5, SCIP, and so just to show some examples, they help to construct now the RNA onto the cavity of PRP8 and stabilize the entire situation. Okay, so having now considering these enormous exchange of these proteins during activation and the substantial remodeling of U2 and U6 RNA, makes it likely that there should be intermediate stages of assembly during activation which undergo only subsets of protein exchanges, and where we may be able to capture also intermediate stages of U2, U6 RNA populations, conformational populations. And in order to purify such intermediate activated stages, we therefore use small molecule splicing inhibitors and showed that these inhibit spliceosome assembly just prior to the mature BACT formation. And then we subjected this to cryo-EM, and Cole Townsend, who worked at the interface between Holger's and my group, was then able to capture two major forms of pre-BACT structures, and which we term the pre-BACT1 and pre-BACT2 structure. So a close inspection of them showed that on the one hand they have certain similarities but also distinctive features. On the one hand, all the most of the U4, U6 proteins are dissociated from both complexes except LSM is still present in pre-BACT1. On the other hand, these NTC, NTR, and RACE proteins are all shared by both proteins, indicating that these are the early guys which associate with the activated spliceosome. On the other hand, the IBC complex and related proteins, they are exclusively located on pre-BACT2, indicating that pre-BACT1 is a precursor to pre-BACT2 structure. A close inspection of pre-BACT2 then showed that almost 22 proteins of the 25 of all proteins which are recruited to the mature BACT are already present in this pre-BACT2, indicating that this was indeed stalled just prior to the final transition to the BACT complex when these proteins, the late-acting proteins, are recruited. Now, what was really striking and unexpected to see was that these 4 B-specific proteins are still present in both pre-BACT structures, and they stabilize the open conformation of PRP8, also in the pre-BACT structures. And this indicates, in turn, that all these 20 and more proteins which have been recruited to the spliceosome during the activation bind while PRP8 is maintained in its open conformation. And this has a major impact in the final U2-U6 folding program of the catalytic site. Now, we were wondering how this traffic could be regulated with all these proteins being exchanged. And a careful analysis of the binding sites of the proteins on PRP8 in the various stages showed here as an example that mutually exclusive protein interactions is a major tool which governs the order of protein recruitment during activation. For example, here in the B complex, we see some of the B U4, U6 specific proteins binding here to the back of the large domain. This is the situation now in the pre-BACT complexes where NTC and NTR proteins bind to overlapping site. That means these has to be first dissociated in order to allow the binding of those. And such a careful analysis of all binding sites in these complexes then provided a basis for the following movie, which provides you with a tentative timing of the coming and going of proteins along the B to BACT transition. So it starts here with the displacement of U4 and U4 specific proteins. Then the early proteins come in, stabilizing the U5' end of U6 RNA. Now the NTC proteins are recruited to the PRP8 RT, and these are important in order to provide now docking site for the 5' domain of U2 RNA, the SF3B associated with the U2 branch site here, because this is still far away, 50 nanometers away from the center here of the PRP8, and therefore this moves now during the pre-BACT1 formation down to this region. And we also include now and transiently the WBP11, if you have noticed this. And now the transition to the pre-BACT needs to displace the LSM proteins. This allows now additional proteins now to really stabilize the U2, U6 RNA here. And we now reorganize also the 5' domain, which adopts now its final position at the large domain. And another protein, HSKIN17, is now intermediately associated here and stabilizes the situation, while the IBC and other IBC-related proteins could also join now. So now we have recruited all these 20 and more proteins to the large domain, while it is in an open conformation of PRP8. And now the final transition occurs once the B-specific proteins are kicked out. And then the large domain can now move downwards towards the NTD, form the closed structure of PRP8, and with it, all the other pre-assembled 20 proteins move together with the large domain without changing their interactions with the large domain. And we create now the binding site for the last 4 late-acting BACT proteins, which finalize now the complement of the BACT structure. And the last remodeling is just the stabilization of domains which have been around but which were not yet stably attached. Okay, so this is now the assembly pathway of the B to BACT transition, and let's now look into the U2, U6 RNA remodeling during these stages. We start, of course, initially again with the B complex. The U6 RNA, the U4 RNA has to be displaced. And what we observe now first in the pre-BACT1 structure is a secondary structure element. It is the ISL of the U6 RNA. And this is now already docked to its final site, which I showed you before in the mature BACT complex, down here at the NTD domain. But it has not the final 3D structure as in the mature BACT complex. It is a more elongated, inactive conformation due to the stacking in of these nucleotides which in the mature complex are bulged out. And this is probably facilitated by the transient interaction of this WBP11 alpha helical piece, which docks and facilitates binding here, and on the other hand prevents the formation of a premature final structure of the ISL. And now, during the transition through the pre-BACT, we now detect the next secondary structure element, and this is the U2, U6 helix 1b. And this is now docked again at its final position, which we have mapped in the mature BACT complex, namely at the helical bundle domain here, and not only the helix 1b, but also the entire U2 RNA, which is supposed to form at the end U6 1a helix, but it cannot do so because the nucleotides which would have to base pair are stretched out in this RNA piece here because we still have the open conformation of the PRP8. And so these nucleotides, which are juxtaposed in the mature structure, are 2.5 nanometers away. So that means the concept is the following. So we keep the open conformation of PRP8 for a while to allow now the secondary structure element formation, such as the U6 ISL and the helix 1b, which can now dock already to their final sites, and to allow the other proteins like WBP11 and HSKIN17 to stabilize intermediate conformational stages. And now we turn into the final structure. So we move the large domain towards the NTD. And in this way, we move the U2 RNA towards the ISL. And this shows now that we can form the loop of the U6 1a loop, which can now base pair with the U2 RNA. And while the large domain is moving towards the NTD, we restrict the open space inside this cavity. And in this way, we facilitate, in other words, the final conformational state of the U2, U6 RNA. And this is indicated in this movie, so where the large domain moves downwards together with the U2 RNA, this folds the U1A loop, and this can now form the U1 helix here. And as the proteins are also moving together with the large domain into the cavity, they will certainly also facilitate now the final formation of this U2, U6 catalytically active conformation of the active site. Okay, so if you give me 1 minute, I just would like to make use of this catalytic site. So this is now a bird's eye view of the BACT structure, where the U2 proteins are still bound to the branch site U2 helix here. So we have to remove all the proteins by a PRP2 RNA helicase. And this has to be moved now towards the catalytic site. And that is shown in this movie here. So the branch site helix moves towards the active site. At the same time, the RNase H domain moves to the forefront and provides the docking site for the extended part of the branch site helix. And this allows then to dock the entire branch site helix into this cavity of PRP8. And we have now carried out the step 1 formation, what you can see here in the high resolution structure. Here we have the branch structure. This is the last nucleotide of the cleaved exon 1, which has flipped back from magnesium 1 to prevent reversibility. And this wouldn't have happened without including additional proteins during the B star to C formation. And these are 10 proteins, and the 3 proteins which are most important are shown here, the YJU2, CWC25, and they together now push the branch site helix into the catalytic center so that the 2' hydroxyl can now attack the 5' splice site, and this results then in the C complex in which the BER2 is now on top of the world, and the next PRP16 RNA helicase has to act to remove these step 1 factors, to remove the branch structure from the catalytic site, and the 3' splice site can dock, and we can form the second step. So, and these are the people involved, and the acknowledgement, and thanks for following this tour to the BEACT complex.
Well, I left this field about 10 years ago, and wanted to think that it was a good thing. I may have been hindering progress. Yeah, I mean, this is spectacular, really spectacular, because and particularly, I think there are a tremendous amount of details here, but what one comes across as dazzling, I mean, it took a lot of structures to do this, is the amazing PRP8 structural transformations during this. Yeah. So, okay.
The next dance.
Yeah. Questions?
Can we have one question?
Yes.
Can I have the microphone?
Yes.
That's my... So given this remarkable series of transformations and comings and goings of proteins, what is the overall efficiency to go from the time that the U1 and U2 bind to the actual first catalytic step? Is half the time this gets stuck somewhere along the way, or does it always work?
It took me 35 minutes. No, I mean, certainly this will be highly coordinated in the cell, of course. And so there is a debate how fast splicing may occur. It's in the 30 seconds up to minutes range, depending on the cells which are investigated, depending on the methods which have been used. So certainly many of the proteins are pre-organized already in protein complexes, which makes it easy. And then they are roaming the nucleus so that the kinetics should be fast enough. And of course, you have many sites where you can interfere and regulate the situation in terms of alternative splicing.
Extraordinary. And yet it makes me wonder, how could this have evolved? Has anybody looked in some kind of cell where there are spliceosomes in action, and yet this process is somewhat simpler?
There are some beasts where you will find some remnants of U6 RNA or PRP8, but it is not, to my knowledge, not really clear that these follow the normal splicing pathway. So the S. cerevisiae yeast has much less in terms of proteins, but other yeasts then have counterparts to most of these specific proteins. So it probably S. cerevisiae has lost something what was early on. So the search for the minimal spliceosome is still going on, I think.
Hi, Reinhard. This is a real tour de force. Just a thought experiment I ask. If you were to do this entire series of experiments in a different intron, a different substrate, how much would be different, and what's your expectation? You know, different proteins, or do you think, I mean, obviously the general principles are there, but how much variation would you expect?
We have done this with quite a variety meanwhile. Also with constructs where we have very long introns, up to 2 kb, which we can now splice in vitro. And you come up, these are viral pre-messenger RNAs, and still we have the same set of proteins. So from all pre-mRNAs which have been used so far and structures have been published, this looks very much the same.
We should move on, John, because
What?
we should go to next week.
Yeah, okay, we should go on. So, I just wanted to say one thing that connects us somewhat to the discussion yesterday. So this structure is enormously complicated, and it's distinguished from the ribosome that it did not evolve probably until the advent of eukaryotes, and the consensus now is that's about 1.7 billion years. So it it's not a fundamental thing, and it used all sorts of bits and pieces that had evolved during that time. And to many of us who looked at those pieces, they were baffling. Well, I mean, he's really starting to understand this. So it's I think there's something profound about making complicated structures when you in fact have a huge number of possible bits and pieces to put into it. Okay, so are you ready? Yes. Okay. So I don't know. Unfortunately, Shigang couldn't come as Tom had explained because of our State Department, and I'm aware. But I think we're very lucky to have her, Michelle Wong, to give her a talk.
Good morning, ladies and gentlemen. I am Rachel Wang from Westlake University in Hangzhou, China. So first of all, I would like to thank the organizers for the invitation. And to be honest, I'm really honored to be here to get the opportunity to meet and exchange ideas with all of you in person. And what makes me feel even more privileged is to be introduced by Professor Abelson and to be arranged to be present my work after Professor Stites and Professor Luhmann. All have been my role models ever since I was a PhD student. So very excited. So let me get started now. And my topic today is beyond the major splicing pathway. Actually, the presentation today for me is a 10 years long story. So let me get started. So in this cartoon diagram of the central dogma, genetic information in DNA is first transcribed into messenger RNA and then translated into proteins. However, in eukaryotic cells, as Professor Luhmann just talked about, an additional and significant step, pre-mRNA, is required. This process, which was discovered over 40 years ago by Professor Sharp and Dr. Robert independently, occurs at 3 conserved sites within the intron through 2 esterification reactions that are carried out by the spliceosome. Actually, the spliceosome is a super huge complex that contains hundreds of components, and meanwhile is highly dynamic. As you can see here, the spliceosome needs multiple orderly steps to assemble and accomplish the removal of one intron. The 2 features above of the spliceosome had made it very challenging to study its working mechanism for decades. And its highly dynamic conformation made it even more difficult to capture the high-resolution structure of an intact spliceosome, which is the most straightforward way to uncover the molecular mechanism of pre-mRNA splicing. So 10 years ago, 2013, the first year of my PhD, I just entered Professor Yigong Shi's lab and started my research. Back then, there were two approaches. One is high-resolution crystal structures of individual components and subcomplexes. The other is EM studies with moderate resolution. So in order to get the high-resolution information, I chose crystallography first. And because none of the building blocks of the spliceosome contains an active site ready for the splicing reaction, we needed to work on the fully assembled intact spliceosome. So to get the sample, we attempted many strategies, and finally I chose to tag the component CDC5, trying to purify the intact spliceosome directly from fission yeast cells. Actually, after many months' efforts, I got the sample, but the yield is too low to allow crystallization. So I persist and improve the yield. And after culturing maybe thousands of liters of yeast, I thought I was very close to getting the crystals, but never succeeded. But fortunately, there was a resolution revolution in cryo-EM in 2013. So encouraged by Professor Yigong Shi, I put my sample under EM, and for the first time, I saw my beautiful spliceosome. It was indeed a very good sample for cryo-EM. So using this sample, we pushed the resolution of the intact spliceosome at that time from nanometers to 3.6 angstrom, which allowed us to atomic modeling the spliceosome. Therefore, in spring 2015, we built the first atomic model of the intact spliceosome, which contains 37 proteins and 4 RNA molecules. Actually, I still remember the stunning moment and tremendous excitement of viewing its spliceosome structure for the first time. And the most exciting observation in the structure is we clearly saw the conformation of the active site. It's composed of intramolecular stem loop of U6, the helix 1 of U6, U2, U6 duplex, and loop 1 of U5. 2 magnesium ions, which was supposed to catalyze the splicing reactions, are coordinated by the phosphate group from this green zigzagged U6 snRNA. And this RNA-based active site is harbored by a positively charged cavity of PRP8 protein. And over the next 4 years, together with Dr. Ray Bai, who is also in the audience today, and Dr. Chuangye Yan, we captured the atomic structures of nearly all the key intermediate structure of the yeast spliceosome. Our colleagues and friends, Kiyoshi Nagai, Reinhard Luhmann, and Ray Zhao also contribute their structures during this time. And with these joint efforts, almost all the major functional states of human spliceosome were also visualized at near atomic resolutions. So based on all these structures, we can faithfully describe how the spliceosome accomplishes pre-mRNA splicing in atomic details. So beyond this, what conclusions can we draw from these structures? First, with protein stripped, we can clearly see the conformational changes of RNA elements. We can see BPS and 3' splice sites are docked into the active site one by one, and during splicing, the coordinated metal ions catalyze the nucleophilic reactions. So this speaks for the metalloribozyme nature of the spliceosome. In addition, although both are ribozymes, the spliceosome is quite different from the ribosome. For the ribosomes, about 60% of the molecular weight comes from RNA, but for the spliceosome, only 10%. So the spliceosome is a protein-directed metalloribozyme. And actually, the spliceosome have a few unique features. First, as we can see from this animation, the components of the protein components of the spliceosome undergo incredible compositional and conformational changes during the splicing cycle. They play indispensable roles in promoting this process. And the scale of the remodeling events is beyond our initial imagination, especially before the spliceosome is activated. This activation step of the spliceosome from pre-B to B-act states. Analysis of the three structures indicates such drastic remodeling events occurred at this step is in order to let the splicing active site form and meanwhile transfer the reaction groups which are needed by the first splicing reaction towards the active site. And second, despite the large-scale structural rearrangement, once the active site is formed in B-act complex, it remains invariant throughout the whole splicing process. This is maintained by a rigid core formed by 21 components of the spliceosome shown on the right. Third, actually as a result of the active site, just so I talked about, the half of the spliceosome, that half, the right half, is inaccessible during the splicing reaction. And the other half, the left half, is accessible for the flux and movement of the dynamic component of the spliceosome to facilitate the substrate delivery. And fourth, all these dynamic processes are ultimately driven by a conserved set of RNA helicases through ATP hydrolysis. Actually, they are always located in the periphery. Fifth, in the spliceosome, some components are unusually more conserved and important. For example, the S-pumping protein SPP42, which hosts the splicing active site in the heart. This multi-domain protein displays almost identical structure and about 70% sequence identity with its functional orthologs in S. cerevisiae and humans. Intriguingly, its finger's palm and thumb domains are structurally homologous to the group 2 intron maturis LTRA in the bacteria L. lactis, which lacks the real pre-mRNA splicing. Last but not least, each major functional state of the spliceosome contains multiple conformationally and compositionally different substates. Professor Luhmann reported 8 substates in human B-act complex, and we also observed multiple states in the human B-act and human C complexes. despite these major conclusions, actually there are numerous questions regarding the function and regulation of the spliceosome to be addressed. But as I start my independent lab 2 years ago, I decided to take on more important challenges. In this case, I'd like to tackle the U12 type splicing or minor splicing. This pathway is responsible for the splicing of a set of non-canonical introns and is only present in higher eukaryotes and remains to be poorly understood. You may ask, in what aspect is minor splicing different from what I just talked about, the major pathway? In fact, minor splicing have several unique features. First, it's rare but essential. Although the minor class intron usually only account for less than 0.5% of all introns in any given genome, they are highly conserved and present in genes that are essential in growth and development. And second, it's quite interesting. For mysterious reasons, minor class intron always appears once and flanked by major introns in a multi-exon gene. Maybe there is a regulation mechanism here to limit the expression of these genes and or affect co-transcriptional splicing, since U12 type splicing is less efficient in cells. So we need to work on that. The minor splicing is executed by the minor spliceosome compared to the well-characterized major spliceosome shown on the upper part. The minor spliceosome is 100 less abundant and uses 4 distinct snRNA, except for the U5 RNA. U5 is shared by the 2 spliceosomes, and the other 4 snRNAs in the minor spliceosome have limited sequence similarities to their major counterparts. So even though the splicing mechanism of minor splicing is likely to be the same as major splicing, the minor spliceosome uses a different set of snRNAs, and they must contain a different set of proteins. So how can the 2 different sets of protein and RNA components execute generally similar chemical reactions in cells? The ultimate way is to study by biochemical and structural approach. So that leads to our approach. First, we must have an assay to monitor the splicing of minor introns specifically and efficiently. And using this assay, we should be able to assemble the functional minor spliceosomes and visualize them by cryo-EM. However, this assay is not that simple. It's very challenging because if I make a comparison here, compared to ribosome, the amount of the spliceosome is just 1/10, and the abundance of the minor spliceosome is 1% of major spliceosome. So the quantity of the minor spliceosome is extremely small, and so it's very difficult to obtain the minor spliceosome, and it's also very difficult to monitor the minor splicing process. So it's very difficult to study. However, thankfully, thanks to Professor John Stites, who was supposed to be here today, nearly 30 years ago, the Stites group already established the minor splicing assay. So we reproduced her results, and actually in the last several years, our my colleagues, especially Dr. Ribai in the audience, and I put a lot of efforts to improve the assay and to study the minor spliceosome. So given the various splicing efficiency of different pre-mRNA substrates, we redesigned a new substrate and performed the in vitro splicing reaction using HeLa nucleic extract. And because it's very difficult for us to obtain P32 in Hangzhou, where we live and work, we use RT-PCR to detect the spliced mRNA product. And to test the specificity, we depleted either major or minor class snRNAs and by the endogenous RNase H. And we can see here, this new substrate can be specifically spliced by the minor spliceosome, although the efficiency was low, with maybe less than 10% spliced. So afterwards, we spent major effort to improve this assay, and now the splicing efficiency is quite high, providing us more convenience to perform more biochemical studies. So with an optimized assay, we try to capture the assembled minor spliceosome.
So it's my pleasure to introduce Professor Tracy Johnson from UCLA. She is an RNA scientist, as many of you know, first became dedicated to RNA splicing in John Abelson's lab, and then is currently working on the very fascinating question of how transcription and splicing are coordinated, because a lot of splicing, perhaps most of it, is co-transcriptional. And so splicing can affect the transcription, and transcription can affect splicing in both directions. But she's also well known for her innovative teaching methods. In fact, she became an HHMI professor to fund her creative teaching approaches. And then she's also, perhaps sadly, an administrator at UCLA where she acts as a dean. So Tracy will introduce today's session. Thank you.
Thank you, Tom. But I will say, first and foremost, I am not an administrator. I am an RNA biologist, first and foremost. So, but thank you so much for really the honor of having the chance to chair this session for many reasons, including being able to chair right after John, who was not only my mentor, but someone who constantly fosters my love of RNA. And I was just thinking about what this community is and what it's meant. And I was reminded that when I was a graduate student, I went to a Nucleic Acids Gordon conference, and a lot of the people who are here and who are giving talks and who have given talks were at that conference. And I remember I was part of the transcription world at that meeting. And I remember looking over at the RNA people and the conversations and how dynamic they were and the collaboration and the innovation. And I thought, you know, someday that's the community I want to be a part of. And so being here today really feels like home. And I would also say that, you know, part of that community were two people who unfortunately aren't here. Joan, whose student gave a beautiful talk this morning, but it's hard to imagine the RNA world without Joan, and also Christine Guthrie, who we lost recently. And so thinking about what does it mean to be part of this RNA community, I think that this meeting has really illustrated the beauty of that. And so I also want to thank you, Tom and Diane, for making this possible. It's really been a wonderful, wonderful chance to get together. So in the last day, we've talked about RNA from the very beginning and its ability to initiate life. And so this, you know, I think I'm remembering one of the questions that Harry asked during his talk, which is, you know, what is the role of the peptides given the critical function of RNA? And one of the answers, of course, is that it helps to guide the structure and the function of those RNAs. And I think this session is going to beautifully illustrate that collaboration between RNA and proteins. And so we're going to have an opportunity to get a tour of the functions of RNAs, including the critical role of RNA turnover through three terrific speakers. So we'll start with Brenda Bass, who will talk about the role of RNAs in antiviral response and the collaborating proteins. And then work from Kelly Nguyen, who's going to tell us about the role of RNAs and proteins at the end of chromosomes. And then Alina Conti, who's going to talk about the role of RNA protein complexes in the transport and the stability, turnover of RNAs, and I think make some larger principles about RNPs, which it's going to be really exciting to hear. So we'll start with a talk from our first speaker, Brenda Bass, who has really had a career of beautifully looking at the role of double-stranded RNAs, from ADARs and RNA editing to the work that she's going to talk about today with double-stranded RNAs and RNAi in antiviral response.
So first, Tracy, you always have very inspirational comments, and those were beautiful. I'm really grateful to be at this incredibly special meeting, and thank you to everyone who has been involved in that. I think that you all know, and Tracy hinted at, that long double-stranded RNA is my favorite molecule. And not many can say, but I can, that it's pretty certain that this molecule was in the RNA world, at least the advanced RNA world that was replicating. So when I first started thinking about long double-stranded RNA, when I was a postdoc, the only time we knew that there was long double-stranded RNA in a cell was after an infection by a virus. And it was clear after infection you could find double-stranded RNA in the cell that matched the viral sequence. To an immunologist, this is a pathogen-associated molecular pattern, or PAMP. It's bound by cellular double-stranded RNA binding proteins to elicit an immune response. Over the first decade or so of work in my lab, we found that our own cells, all animal cells that we've looked at, encode and express long double-stranded RNA that, at least at first glance, looks very much like viral double-stranded RNA. So we think a lot about, in my lab, how do we know the good from the bad? How do our cells tell? So as I start this talk, I need to remind you that in vertebrates, there are proteins called RIG-I. Can you guys see that pointer? Okay, except for those that are colorblind, maybe. In vertebrates, you have RIG-I-like receptors that recognize viral double-stranded RNA to trigger an interferon pathway. In vertebrates, things like C. elegans and flies. I think that might be good. I was making sure I wasn't attached to anything. Okay, cool. Thank you, Rachel. So in invertebrates, things like C. elegans, flies, there is no interferon pathway. And instead, it's dicer that binds and cleaves the viral double-stranded RNA to give viral siRNAs that target the viral transcripts through RNAi. So some of you are probably aware that the mammalian protein RIG-I recognizes and it distinguishes viral double-stranded RNA by the di and triphosphate at its end. And RIG-I knows that that's not a cellular double-stranded RNA, which would have a monophosphate or a cap. And my lab has determined that invertebrate dicers also distinguish termini. For example, dicer can distinguish a blunt double-stranded RNA, and we think of this as a viral mimic. And dicer knows that that's not a cellular double-stranded RNA, for example, watch closely, that might have a 3' overhang, like a microRNA. I've shown all of these proteins as red ovals because they have a highly conserved helicase domain of the same family. And this makes us think that this helicase domain functioned in antiviral defense in a common ancestor. As we've gone more and more into these studies, we've become really interested in how the innate immune response evolved. One thing I haven't told you, but I'm sure you know, especially Phil sitting over there, humans, we have dicer, but it's very controversial whether human dicer is involved in antiviral defense. And it's a rather wimpy enzyme. And we hope that by studying invertebrate dicers, which are quite robust, someday we can turn the human enzyme into something that can efficiently target virus. So I'm going to be talking about at least 3 different dicers today. As I talk about them, I want you to think about how dicer has evolved as the innate immune pathways diverged. And I need to remind you that dicer is not a housekeeping gene. It has been bombarded by viruses trying to cripple it throughout its evolution. Okay, dicer is an L shape. It has a highly conserved helicase domain, and you're looking at the 3 subdomains. In the middle, it has 2 ribonuclease 3 domains. Each cleaves 1 strand of the double-stranded RNA. At the very top is the PAS and platform domain, and it was proposed many years ago, some of these people are in the audience, that by binding the end of a double-stranded RNA, this allows measuring to the RNase 3 cleavage site, and this explains why siRNAs and microRNAs are 2 turns of the helix. So a really confusing thing is that despite the human enzyme having a very conserved helicase domain, it has never been observed to use ATP. In the literature, the model that exists, and this is animated by my colleague Janet Iwasa, is that the end of the double-stranded RNA comes up to the platform PAS domain, swings in, cleaves, the RNA is released, ATP-independent distributive reaction. This is what everything points to for the human enzyme. And this confused us to no end about a decade in our studies of the Drosophila enzyme, Drosophila dicer 2. We knew it required ATP. We knew it was processive. And it was only when we teamed up with my colleague Peter Shen, a cryo-electron microscopist, when we got a structure of Drosophila dicer 2, animated here again, we realized, in this molecule, double-stranded RNA is entering the helicase domain. It's unwound and clamped. You get little pieces as it passes the RNase 3 nuclease site. There is a measuring to give the mature siRNA species. It's processive. This would be a great way to chop up viral double-stranded RNA. And I want to make sure everybody knows this enzyme I'm telling you about, Drosophila dicer 2, is necessary for flies to fight viruses. What you're looking at here are flies that have been infected with Drosophila C virus. Wild-type dicer, you see nice viral siRNAs. You make a mutation in the helicase domain. The flies cannot fight virus. Okay, what has happened to human dicer? This is a project that has been done by Deji Adaranmu. Some of you have probably met him already. He defends his thesis on Friday. And what Deji decided to do was a phylogenetic analysis of the helicase domain as well as an adjacent domain, called DUF283. What Deji did was called an ancestral protein reconstruction. This is sort of exactly like it sounds. It's a little bit Jurassic Park. You reach back and predict the sequence of the ancestral proteins. You make them in the lab and test their activity. Deji wanted to do these studies because he wanted to understand what happened to human dicer. Did it lose ATP hydrolysis, or did everything else gain it? In an ancestral protein reconstruction, you gather sequences, and Deji went to NCBI and got many animal protein sequences. You then do a multiple sequence alignment. You create trees to tell you which proteins are related to which others. And then, the important part is people in this field have developed algorithms that allow you to look at the nodes of your tree and, again, predict the ancestral protein. So you can order the DNA and make it in the lab. This is just to remind me to tell you that we know our trees are high confidence. This is an example of one of Deji's trees. This is the ancestral dicer. If you put a number on it, you would say, well, maybe 700 million years ago on this tree. There was an early gene duplication. Most everything we think about in animals, oh, you can barely see it, but comes from dicer 1, this pink shaded region. And in fact, only arthropods kept both dicers. And I've been telling you about the modern day Drosophila dicer 2 that has an antiviral function, but even modern day flies have a second dicer. It's dedicated to microRNA processing. So Deji made proteins for every node you see here in black. He ordered the DNA, expressed the proteins in the lab. And we're going to look at these by ATP hydrolysis assays. And what I'm showing you here is actually an assay of the modern day Drosophila dicer 2. These are thin layer chromatography plates, P32 ATP going to ADP. And I want you to notice the blunt double-stranded RNA, the mimic of the viral double-stranded RNA, stimulates this reaction better than a 3' overhanging double-stranded RNA. Okay, I'm going to show you some primary data from Deji. Again, we're looking at his tree, most ancient to the left. What you see is that even without double-stranded RNA, you have a little bit of ATP hydrolysis. It is stimulated by double-stranded RNA, but you don't see any difference in the stimulation between blunt and 3' overhang. Importantly, we've got our first answer, right? Because we see ATP hydrolysis in the ancestor, the human enzyme, vertebrates had to lose this activity. If you look up here at the deuterostome node, now these deuterostome are things that look like us or starfish. You can see that, yes, deuterostomes do hydrolyze ATP, and importantly, now we can see that blunt double-stranded RNA stimulates a bit better than 3' overhanging double-stranded RNA. Again, the blunt is the viral mimic. And finally, drum roll, we go over here to the vertebrate ancestor. No matter how much protein Deji put in these assays, no ATP hydrolysis activity. Deji, of course, did multiple assays, and he has rates for these reactions. In general, as you go from ancient to modern day, dicer gets slower and slower. Deji also determined binding affinity for double-stranded RNA. Ancient binds pretty well to double-stranded RNA, but again, we're losing affinity as we go to the more recent organisms. Okay, so I want you to remember that our goal is to rescue the human enzyme so it can function efficiently in antiviral defense. If we had started with the modern day organisms, it would have taken us a long time to do this rescue. Drosophila dicer 2, humans, only 25% identical. So figuring out which amino acids we should change would have taken a very long time. Fortunately, now we have this deuterostome ancestor, and it is 64% identical to the vertebrate ancestor. 200 amino acid differences. That's better than where we were before, but still sounds like a lot. So Deji decided to get some mechanistic insight that might help us focus on where the amino acids were that were causing the vertebrate ancestor to lose its activity. And I want to just one more time point out that this is a really big mystery. If you look at alignments of modern day animals, and here I'm pointing to the Drosophila dicer 2 and the human enzyme, and I'm pointing to the motifs that are known to be important for ATP binding and hydrolysis, and they are highly conserved. So if it's not these motifs, what is it that has led to the loss of activity in the vertebrate ancestor? So what Deji decided to do was some Michaelis-Menten kinetics. It was a very, very good idea, entirely Deji's idea. And what we're going to look at here, of course, is the ATP binding pocket, saturation. So you're seeing normal velocity versus ATP concentration, ancient versus more recent. What you're seeing in red is no RNA. Blue is with double-stranded RNA. You should be confused. And I see some people nodding, they are confused. What is happening over here? It looks like the RNA isn't helping at all. But if you zoom in on the ATP concentrations that are physiologically relevant, now you see, okay, the RNA is helping a lot. And if you look at the Km values, you see that double-stranded RNA is really lowering the Km. It's allowing the ATP binding pocket to be saturated under physiological conditions. Okay, when we saw this, it started making us think, well, maybe vertebrates lost the ability to couple double-stranded RNA binding to ATP binding. And this was interesting to us because our lower resolution structure, as well as these nice structures that have come out in the last year, show that the helicase domain of Drosophila dicer 2, when it binds double-stranded RNA, clamps around the double-stranded RNA. And what this does is to bring closer the HEL2, HEL1 domains, the REC-A domains. When double-stranded RNA binds, the ATP binding pocket is created. So we started to think, maybe this is the problem with the vertebrate ancestor, and we got a little bit of fuel for the fire, because when Deji sent his modern-day Drosophila dicer 2 and all the active ancestral constructs, he got a confirmation from AlphaFold that had the REC-A domains in the right conformation. When he sent to AlphaFold the human enzyme or the inactive vertebrate ancestor, those HEL2 domains were not in a correct conformation. Okay, can we use this to try to, this mechanistic insight, to try to rescue these, the vertebrate ancestor? So the first thing Deji did, and I think Deji's probably up to vert.10 now, but I'm showing you data for vert.1 rescue and vert.7. And we're going to first look at those motifs that I said were really highly conserved in the ATP binding pocket. Even though they're highly conserved, you can find things that differ. And so DEJI corrected 20 of those sites. Both vert.1 and vert.7 have those corrected residues. And then only in vert.7, he looked outside. He looked at things that he thought might be involved in the conformational change and also things that might be involved in double-stranded RNA binding. And it's hard to see, but in this very last row here is vert.7, and he's rescued some of these amino acids, and this was the important part, and vert.7 was rescued in its ATP hydrolysis function. So just want to remind you, the vertebrate ancestor, no ability to hydrolyze ATP. Now, with these additional 40 amino acid changes, this enzyme can hydrolyze ATP. If you look at the KM with double-stranded RNA for vert.7, you know, we've lowered the KM, but we're not quite there. We haven't especially created the very efficient enzymes that are allowed in these ancestors. So it was really surprising to us and unexpected to find that what has happened in the vertebrate ancestor is that it appears to have lost the ability to couple double-stranded RNA binding to creation of the ATP active site. Make sure I'm doing okay here. So in just, yes, good, 5 more minutes. In these last few minutes, I just want to say a few things. So once you start thinking about, you know, how do modern-day dicers reflect the evolution of antiviral defense, it's very hard to stop. And we've been thinking a lot about C. elegans dicer. This was actually the very first dicer that my lab ever studied. And despite numerous attempts, until recently, we have never been able to purify this enzyme in an active recombinant form. And about a decade ago, Thomas Duchesne and his laboratory found that when C. elegans dicer functions in antiviral defense, it does so in collaboration with a double-stranded RNA binding protein called RDE4 and a third protein called DRH1. And intriguingly, DRH1 is actually an ortholog of RIG-I. So now we have 2 helicases functioning in antiviral defense. Claudia and DEJI first collaborated on purifying these 3 proteins together, and it worked quite well. Claudia has taken this project forward, and she will be at her poster today to tell you all about it. The punchlines are that the complex is active, so for the first time, we do have active C. elegans dicer. She has a cryo-EM structure of the whole complex and a higher resolution structure of the RIG-I-like homolog DRH1. And an important thing for me to say is that it's very surprising, but in this complex, DRH1, the RIG-I homolog, seems to play the major role in ATP hydrolysis over dicer. And I want to end on comparing some of the enzymes I've told you about. I started out telling you about the human enzyme. You know, it seems to have given up on dicer for the immune pathway, and it lets the RIG-I-like receptors couple ATP hydrolysis to antiviral defense. On the other extreme, we have Drosophila. Drosophila has no RIG-I-like receptors. Dicer is the main player in the show and couples ATP hydrolysis to antiviral defense. And then we have C. elegans in the middle. DRH1 seems to be the main protein that's providing the motor function, but dicer still can hydrolyze ATP. And I'll go to, so what I've told you today is that we've used ancestral protein reconstruction to resurrect ancestral dicer helicase domains. I've told you that the common ancestor of animal dicers could hydrolyze ATP, indicating that the vertebrate dicer lost this activity. As dicer lost the arms race with viruses, it became less efficient at ATP hydrolysis. Michaelis meta-analyses suggest this is a KM effect. And we've rescued ATP hydrolysis in the vertebrate ancestor, but we want to go a little further to get a more efficient enzyme. And I will close by thanking the stars of this show. Deji Adaranmu has done all of the ancestral protein reconstruction. Some of you probably were lucky to see his poster yesterday. He and Claudia worked together on the early part of the C. elegans study, but Claudia has spearheaded all of the subsequent work and has a poster today. And Helen Donalick, a former graduate student, assisted Claudia with the structural work, and of course, she has been snatched up by Thermo Fisher. My longtime technician, right-hand man, Joe Arascavage, helps everybody with everything. I'm very grateful to my funding, and I want to note that the other scientists that I share a transformative R01 with have really helped shape my ideas about innate immunity. And I will stop there, and thank you.
Thank you, Brenda. That was really beautiful. Any questions?
Great talk. I thought this was like some wonderful evolutionary biochemistry. Really made me think. I was curious, kind of in a larger picture, what you think the selective pressures or lack thereof over time were that caused us to lose this ATPase function in dicer. Do you think that it was, the emergence of this kind of complementary, redundant, perhaps more efficient RLR system? If not, what so? And then if so, how much hope do you think that there is of basically rescuing this, having any improvement in antiviral?
Yeah, boy, that's a great question, and it's multiple questions. But I will tell you first that, viruses, of course, did not want dicer to knock them out. So at some point in time, I would say the human enzyme gave up, said, okay, let's use those reg-I-like receptors. The other thing that I guess I will say to you, I'll add a little complexity in that there's no evidence that human dicer can ever thread and act efficiently to chop up a virus. But in germ cells and stem cells, which are very sensitive to interferon, and it's detrimental to them, it does look like dicer does help in antiviral defense. So not efficiently, but it's there. And the other thing that it's fun to think about is, okay, reg-I-like receptors took over invertebrates, but microRNAs were just proliferating in vertebrates. And so that was another thing that probably said, okay, we're going to specialize that vertebrate dicer for chopping microRNAs.
So obviously you know this better than I do, but ADAR is the other way to take care of double-strand RNA. So if you look through this evolutionary relationship, do you see a corresponding evolution of ADAR in level of activity and type of activity?
Yeah, we have talked about that. We have not done that. It would be really fun to look at that. One of my TR01 collaborators, Nels Elde, did spend some time looking for regions of positive selection on ADARs and has not found anything convincing yet, but it's a wonderful thing to look at.
Does the C. elegans structure suggest that the reg-Is might associate with dicer in humans, and is there evidence for or against them?
Yeah, we think that's a really cool idea. I mean, because you see DRH1, this is a cartoon. You guys will get the more anatomically correct picture from Claudia's poster, but Rachel's question is, okay, this is an ortholog of rig eye. Is there any evidence in vivo that this interaction could happen? We're going to keep that in mind. Nobody's ever seen it. However, I will say there's a lot of antagonism between Dicer and the RLR pathways in mammals. And another rig eye-like receptor, LGP2, is known to bind to the helicase of mammalian Dicer and inhibit it, as if the rig eye pathway say, hey, don't chop up my double-stranded RNA. I'm going to use it for signaling. But it's a fun idea to think about. And Jamie?
So I'm intrigued by the fact that you kind of stop at this part of the phylogenetic tree, and I'm thinking about what's going on with the fungal dicers, and this, especially thinking in light of this complex you're seeing on the right.
Yeah, Deji actually rooted our trees with fungi. And so you can play this game with so many different animals. What's happening in fungi? I'm very curious about mollusks now. Deji, do you have anything to add about fungal dicer and whether, there he is. Do you? You're defending your thesis on Friday. I mean, you get.
So I'll say, not a lot has been done with dicer and antiviral defense in fungi, but in plants, most plants actually have 4 dicers, and one of them is specialized for antiviral defense and has helicase function. So we kind of think that this function is more ancient than animal evolution, but no one has really looked closely in fungi specifically. So yeah, thanks.
Very good answer.
We have time for one more question.
Okay, there's somebody in the back. Yes.
Your talk reminds me when a few years ago I was looking to some dicer CRIP data sets for binding sites for dicer, and surprisingly, precursor microRNAs was the not most frequent target. Actually, it was not even in the first 3.
So sorry, the microRNAs were the most. No, we're not.
No, we're not.
Thank you.
So the most frequently bound targets, according to some PAR-CLIP analysis, was the tRNAs and the double-stranded regions of tRNAs. So maybe do you know anything about physiological role, if this binding could have any physiological role, or it was just a method artifact?
Yeah, I would love to talk to you later more about this. I do know that people do see tRNA fragments that are functioning in silencing. And I think I remember that Dicer is thought to make some of those tRNA fragments. Is that, if anybody knows differently, tell me. But yeah, I would love to talk to you more about that. That's interesting.
What a great way to conclude. Thank you so much, Brenda. So I'm really excited to introduce our next speaker, Kelly Nguyen. And I think some of us may have first been introduced to Kelly at a meeting in which she was working in Kyoshi Nagai's lab and showed these beautiful triple SNRP structures in the spliceosome. And so it was exciting to see how far we've continued to go in understanding the spliceosome. But I think that early beginning gave us a hint at the gorgeous work that Kelly does. And so we're excited to hear how she's thinking about and solving the structure of another really important RNP, the telomerase. So thank you so much.
Thank you so much, Tracy, for the very kind introduction. And first of all, I would like to thank the organizers for giving me this opportunity to be here. I feel extremely privileged because it's rare that, so many of my scientific heroes are all in the same room. So yeah, amazing. So today I'm going to tell you about the work that we have been doing the last few years on trying to visualize human telomerase, and this is the molecule that's shown in the right. And so to start with, I'm going to give you a little introduction what we study and why telomerase is important. So my lab at the OMB studies this region of the genome called telomeres, and these are the region located at the ends of the chromosomes that you can see here in this metaphase spread, these dots that are at the ends of the chromosomes. Now, if we look a bit more closely to see how chromosome ends are structured, as you heard from various talks yesterday, eukaryotes generally have linear ends, as shown here. And in most eukaryotes, the telomere consists of G-rich repeats. And in human, this is just GTTTAG, nothing else, so 6 nucleotides. Also, it ends with the 3' end overhang on the G strand. And this can range between 200 kilobases to 100 kilobases in mammalian cells. And what we know is that in mammalian cells, there's a complex called the Shelterin complex that binds to double-stranded DNA at the telomeres as well as the single-stranded region. And together, it forms kind of what we think as a cap at the end of our chromosomes, and therefore protecting the chromosome from degradation and also inappropriate break repair. But now, if you look at this linear structure of chromosomes, it's actually problematic for the ribosome to replicate due to how DNA polymerases work. So due to this, this leads to what is famously known as the end replication problem. So with every cell division, we lose a bit of our telomeres. So as a result, over time, the telomeres shorten. So when it becomes about 2 kilobases or so, and this is known as the Hayflick limit, the cells can no longer divide. So they will undergo growth arrest, geometric instability, and eventually cell death. And unfortunately, this telomere shortening effect happens for pretty much all somatic cells in multicellular eukaryotes. That's most of what's in our body. So now the reason I'm here is because the RNA will come to the rescue of this end replication problem. And this is where it lent a hand via an enzyme called telomerase, which is a ribonuclear protein. So telomerase is essentially a reverse transcriptase, but it's not a usual one, because not only that it contains its own reverse transcriptase subunit called TERT, shown here, it also has an RNA component called HGR in human. And what this RNA component brings is that what we call the template region is shown here, and half of it can specifically recognize the end of our DNA by just base pairing, very simple. And then the other half of the template region is then used by the RT to synthesize the exact GTTTAG repeats. And what is also special about telomerase compared to retroviral RT is that it can do this multiple times every time it binds to a DNA. And this special processivity property of telomerase is what we call repeat addition processivity. And this is absolutely crucial for telomerase activity in the cell. And this also very nicely recapitulates the in vitro in the field by using this in vitro telomerase assay. When you give it a DNA substrate and dNTPs and the GSP32 label, when you run it on a sequencing gel, what you can see is this beautiful ladder of 6 nucleotide repeats being added to the DNA. And not only telomerase is very interesting, in terms of molecular biology, but it's also very important clinically because most of our cells do not have telomerase. But it is actually upregulated in cancers, stem cells, and germline cells, and this allows these cells to divide indefinitely. And this is what we call cellular immortality. And this is where it got the name as the immortality enzyme in the popular media. So basically, what I just told you, that telomerase needs these 2 components to synthesize the repeats. But in the cell, telomerase is actually more complex than just these 2 components. So now let's start with the RNA. As you can, many of these in the audience here obviously need no introduction on RNA secondary structures. So the RNA telomerase is about 451 nucleotides, and roughly we can divide it into 3 parts. So the first part is what we call the template pseudonode domain. It has this template region that's used for DNA synthesis, and a conserved regions 4 and 5. And the RT is known to bind to these 2 domains of the RNA. And you would notice that at the 3' end of the RNA, there's what we call the HACA domain, and that's known to bind various proteins to form what we call the HACA RNP. And it turns out that this family of RNP is not limited to telomerase. It's actually shared with a large family of RNP, including the SNO RNP as well as the SCAR RNPs. And it's present in both archaea as well as in eukaryotes. The archaea version had this simplified RNA that only had a single hairpin structure. And in eukaryotes, the RNA had this double hairpin structure flanked by this highly conserved H and ACA boxes. And what we know is that on each of these RNA hairpin, there's a tetramer of this carinin, NOP10, and HP2 and GAP1. So the purpose of today's talk, you don't need to remember these names, but just keep in mind the tetramer of these 4 proteins. And also, some of the RNA also have this cap box in the loop, which can also bind TCAP1, which is a Cahal body localization factor. So the majority of these HACRNP function to convert uridine to pseudouridine in the ribosomal and spliceosomal RNAs. They're very important for the biogenesis of these large RNP machines. So when we started work with telomerase, there's been beautiful structure of single hairpin HACA RNP, and the archaea example is shown here, but there was no eukaryotic HACA RNP structures available. So with this information in hand, so various labs in the field, including my old boss, Kathy Collins, come up with this monomeric TERT HTR model where human telomerase consists of TERT, the RNA, 2 copies of the HACA proteins, and TCAP1. But those in the telomerase field would know that this actually was for many years controversial. Thus, this is among the many models that were proposed to be, was actually in the enzyme. So I was very fortunate to be offered to study this enzyme when I applied for a postdoc with Kathy Collins and Eva Nogales at UC Berkeley. And what we want to do is to take advantage of, you already heard about this cryo-EM resolution revolution in order to determine the structure of telomerase and resolve this issue. And many of you, all of you would have heard that about 10 years ago, our ribosome friends, as well as membrane protein friends, really enjoyed the big gamma resolution thanks to the technology developments in the cryo-EM field. And structures were able to be solved at 3 to 4 angstrom resolution back in 2013. And just to give you a reference where we were on the same year with telomerase, with human telomerase, that there were also a structure of human telomerase, so in the same year. And as you can see, the resolution is 30 angstrom. So as you can see, that this is very far in terms of resolution. So what I'm going to tell you for the rest of the talk today is how we are able to bring this gap down, and now we're almost catching up. So also based on this structure, this will propose to be a dimer of TERT and HTR. So again, controversial. So the first thing we do is actually develop an approach to purify telomerase, because as I mentioned, it's extremely rare. So making telomerase themselves is really, really hard. So the first thing we do is to use an expression system where we only expressing the limiting factors, which is the RT as well as the RNA. And then by transfecting hundreds and hundreds of plates of cells, we were able to get to the quantity in the microgram scale, which then can be used for structural studies. And it's the same here. Now we can actually silver stain the sample and can see a lot of the components that I mentioned before. And then because it's an enzyme, I mentioned that we can do this in vitro telomerase assays. You can see that the reconstitute complex is highly active based on these assays. And really encouraged by this, we then moved to cryo-EM, and you can see the cryo-EM class averages as shown here, and it showed that the structure is very homogeneous in terms of composition, but it's very flexible in terms of conformation. And this is the nice thing about EM, because you can actually collect millions of particles, and then by computational means, you can actually drill down to a couple % of the particles that have homogeneous conformation that allow us to solve the structure to 8 angstrom. So you would notice that this is quite far from 3 to 4 still, but this resolution tells us a lot about its composition as well as architecture. So I'm going to summarize the structure in this slide here. So I'm going to start with what you've seen before, shown here, and then I'm going to redraw it to what we see in the structure. So one of the most beautiful things about seeing this structure is how the RNA actually scaffolding the entire structure, as shown here in these different colors. So all the protein kind of gets stitched together by the RNA. And now let's zoom out of the RNA, focus on the protein parts. So here's the actual electron cryo-EM map. And here I'm coloring the RNA as blue, as you can see here. And then the proteins are different colors. So in the bottom part of the RNA is the RT bound to a DNA substrate. And on the top part is the HACA RNP bound to 2 copies of the tetramer. The first one is here, second one, and then you have TCAP1 just cap the top of the structure. So this nice thing about doing structure is this literally seeing is believing. So you can actually see that this agrees with the monomeric model of telomerase. And the structure also tells us that these proteins are segregated in 2 lobes, as you can see here, and they're very functionally different in terms of what they do for telomerase. So the top part, called the HACA lobe, is important for biogenesis, also localization of telomerase to the Cajal body. Whereas the bottom part contains the reverse transcriptase, so we named this the catalytic core because it's important for DNA synthesis and also recruitment to telomeres. So now the structure, tell us about these composition architecture, but again, this is very far from the 3 to 4 angstrom resolution range I talked about. The question is, how do you improve the resolution? And the short answer is a few things. So first is to apply the latest technologies, collect many times more data, so about 15 times more than what we have. This is equivalent to about 10 days on the microscope, in a straight go. And also more computing. So by doing all of this, we were able to make the big jump to below 4 angstrom. And the work is really done by 3 really talented people in my lab. George is here and giving a poster this afternoon. And I also want to mention that this was done in the middle of the pandemic and across the Atlantic, because we work also with Richard Das, who's here, and also with my old boss, Kathy Collins. So what I'm going to do is going to start with the old structure, because you've seen that before. And it's divided into 2 parts. You have the HACA in the top, the catalytic core in the bottom, and then I'm going to morph it into the new structure. So now we are in the range below 4 angstrom. You can see, and we can see so much more details than we did with the previous structure. And at this resolution, we can build an atomic model. And it was a very fun lockdown project for everyone in my lab. So here is the catalytic core that has the RT as well as the RNA and a histone dimer. And we have the DNA substrate in our ensemble. So the first time we can actually really visualize how the DNA substrate interact with the active site of the RT, as shown here. And because as I mentioned, there was no histone in the previous structure, right? So I will tell you a little bit about that. But by solving this structure, we identify histones as part of human telomerase. You can see here that's them associated with the enzyme. And now, because there's no structure eukaryotic HACA available before our work, so by solving telomerase structure, we happen to also solve the first eukaryotic HACA RNP, and you can see here that's atomic model that we model from the density. And now, in the middle of the 2 structures are quite flexible, so we obtain a low-resolution structure, and by working with Ritu, we are able to model the entire RNA linker between the 2 lobes and allow us to obtain complete atomic structure of the enzyme, consisting of now 12 proteins and 1 RNA. So now there's a lot one can discuss about the structure, but I'm going to kind of focus on one point that was surprising when we solved the structure that couldn't be identified in the previous genetic and biochemical work, and that is identification of the histone dimer. So actually back in 2018, when we published our previous structure, we had this suspicious density that kind of sticking out, and we didn't know what it was. And there was not enough resolution to model, so we just put in the supplemental and thinking of coming back to it later. And thanks to the new resolution, the new structures that had better resolution, so we asked a very simple question. given this density, what is in the BDB will fit in this map. So pretty much search for a large library from the BDB. And the surprising thing that we got is the top hit returns a histone H2B. And that is our density, and histone H2B is here. And you can appreciate that it only fit half of the map. So naturally, we take H2A from the same model that returned the hits and put it into the map, and here comes, it fits perfectly. And that allows us to pretty much redraw what's actually in the human telomerase catalytic core, as you can see now with an extra pair of histones. So you probably wonder, what could they be doing telomerase? And yesterday, Tom gave already an answer to that, because DNA binding proteins can also bind RNA, and this is a perfect example of that in this case. It turns out that the histone binds a conserved motif of the RNA on telomerase RNA, and this is called this conserved regions 4 and 5. It's located here. And this has this three-way junction structure you can see, and the histone binds across two of the stems across this three-way junction. You might also remember from my previous slides that the same part binds to the RT as well, and it binds on the other side of the RT. And what is remarkable is together they seem to shape this stem loop here of the RNA, and this has been shown by many previous work to be essential for the catalytic activity of telomerase. So if it's misfolded or mutated, telomerase is dead. So now, not surprisingly, because it combines DNA, so in some ways it uses the same kind of surface to bind DNA, and it's really interacting a lot with the phosphate backbone of the RNA. You can see in this surface chart diagram. And if you put this, the structure of the nucleosome side by side of our structure, it really binds to RNA the same way as it binds to DNA. So that's quite cool. But further from that, we not only observe RNA backbone interactions, but we also observe some interaction with the RNA bases. So one of such is shown here. You can see zooming in, you see there's this G310 stacked with this R82. So we then can ask, is this interaction important? So what we did was reconstitute telomerase again, and basically reconstitute it with this mutant, as you can see. So we reasoned that we had to delete it because any of the bases can form the same interaction, so we make a delta mutant of this G. You can see it's abolished telomerase activity. And we also see another cluster of base-specific interactions up here, and this guanosine form interaction with this arginine. Again, we do the same thing. We mutate this guanosine to other bases, and you can see it drastically reduce telomerase activity. And another one is the U316 that also interact with the backbone of the histones. And again, we mutate this. And except for alanine mutation, adenosine mutation, the U to G and U to C disrupt telomerase activity. So these interactions seem to be important for telomerase activity in some ways when we reconstitute this in vivo. So now we're really trying to address quite a number of questions following this is still kind of ongoing. So what first is what they're actually doing in telomerase. Is there anything more than just RNA folding? And we also don't know whether there's canonical histones or any variants. And we also consider a possibility that there's a histone chaperone involved. And beyond telomerase, because they interact really just mostly of the RNA backbone, so we also ask if there's any other RNA that's also involved in histone interactions generally. So now the methods we develop can then allow us to go to the next level when we actually study how telomerase is regulated in cells. So most of the time, telomerase is not at the telomeres. And you might remember from the very first slide, at the telomeres is the complex for the Shelterin complex. It protects the end of the chromosomes. And telomerase is often tucked away in the Cajal body in the nucleus. So only at the end of the S phase, the telomerase get recruited to the telomeres by Shelterin itself, and this recruitment is essential for its activity in vivo. So we would like to understand this recruitment process. And among these subunits of Shelterin, there's one that's called TPP1 that's been found by Tom, Kathy, and also Steve Attandi's labs to be important for recruitment of telomerase by direct interactions with telomerase. And also, there's another factor called POD1, which binds single-stranded DNA, has been implicated in this process. And these two has been shown to also simulate telomerase processivity. And this can be, again, very nicely recapitulated in vitro by many labs, and we just kind of repeat these assays on our hands. You can see here, when you have telomerase and DNA, you make the repeats, and you add shelterin at higher concentration, you can see that it makes longer DNA. So at the time, the structure basis of the recruitment and simulation were unknown, so we thought we can try to use our methods to do this. So my postdoc, George, again, who's here, and Zala team up to solve the recruitment complex structures. So again, I just give you the schematic telomerase that has the HACA lobe and the catalytic core. TP1 POD1 binds to the catalytic core as shown here. So we solved two structures, and the first is with telomerase with TPP1 and the DNA, and it's shown here, and that is TPP1, and on the top it's telomerase catalytic core. And the second structure is telomerase with TP1 and POD1 with the DNA, and it looked the same, but POD1 is binding in front of the structure. And what I'm going to show you next is a movie that's kind of hinting at how T1 and POD1 recruit telomerase as well as increasing its possessivity at the telomeres. So here's the first structure. You have all the components of telomerase, and TP1 is at the bottom here. And so we were able to build atomic model for this complex, and you can actually zoom in to see how TP1 recruits telomerase by interacting with these domains of telomerase and using this highly conserved motif that has been identified by Tom and also JK, who is a postdoc in his lab. And the second structure is with POD1, which is a single-stranded DNA binding protein, so it's bind on top of the active site of telomerase. Because it binds DNA, so it can actually resolve more DNA in the structure, as shown here in red. And what we found is that based on the structure, the two proteins together allow telomerase to engage in the DNA more stably. And you can see from this side view that this DNA path coming out of the enzyme. So previously, we see only this part that are here in the active site. So what we think they work by, not only recruit telomerase by TP1, but also stabilizing telomerase on the DNA so it doesn't fall off during synthesis. So because for this work, we collected a lot of data, so we have a really nice byproduct, which is with the structure of the HACA RMP of telomerase, which stays almost the same, but it goes to very high resolution, as you can see here, at 2.7 angstrom resolution. This is currently the highest resolution of any telomerase complex to date. And we learned a lot of cool things about the function of this HACA RMP. So now I'm just give you a little summaries that we 10 years ago, we far from that resolution rank of the revolution, but we're now starting to catch up. So starting from this really, still very informative structures by Donella Rose and Joachim Linger, we're now having to, in the last 5 years, we're able to bring it down to below 3 angstrom. So we're about 1 angstrom away from the current ribosome high-resolution structures. So we're not so far up. And I would like to also mention the work done by Mingley and Julie Feigen on related complexes and show very similar findings to what we found. So with that, I would like to acknowledge a lot of people, and first and foremost, Kiyoshi Nagai, who sadly passed away 3 years or 4 years ago, who's really introduced me to the RNA worlds. And my scientific childhood is working on the spliceosome, as some of you might remember from past RNA meetings. And Kathy Collins and Evan Nogales for this amazing opportunity to join the lab and work on telomerase. And all these wonderful collaborators and colleagues at the LMB, and all my lab members and the people who did the work in both. And you might recognize who's, I have a super postdoc who joined us for sabbatical, Don Rio, so we've really kind of had a really nice time with him. And thank you for your attention.
Thank you so much. It was wonderful.
Thank you.
Hey, Kelly, fantastic talk. So over here, so the question about the H2AB histone binding, is there any chance that there's any histone modification in there? Because obviously the telomere ends are often heterochromatin, so there's specific histone marks there, right? Is there a role for that in the recognition or in the subunit?
Yeah, that's a great question. So it's a shame that we only see the core histones, not the tails when the modifications happen, but we're now chasing up the one of the hits for histone chaperone, and that actually deubiquitinase H2A. So there could be something that bring it onto telomerase that, you know, with our system we're not yet being able to capture. But yeah. Yeah, Joe?
Kelly, it's a beautiful, spectacular. I had a couple questions, which are, how does translocation occur once you do 6 nucleotides?
Yeah.
And in the active site, are the active site residues positioned near the primer, or is it in a kind of awkward catalytic state?
Yeah, it's a very good question. I didn't go into details, but what we capture is the most stable. We use a primer that binds to the active site most stably because it ends with GGG. So it's actually, yeah, so we think it's happened based on this structure alone is the duplex actually quite short, only 4 base pair. And on the other side, it seems to peel away so it doesn't extend like this and then just suddenly jump. But now what we're doing now is actually trap the enzyme sequentially to see actually how the active site actually can do pretty much 6 times before it had to go back. So it's this...
HIV reverse transcriptase that after you add 6 nucleotides of the RNA DNA duplex, you start to get flexibility.
Mm-hmm.
I wonder if something similar is going on.
It's kind of because we were expecting to see more base pair, like longer than what we saw. So we actually see only 6, and the other bases that can actually hybridize doesn't form base pair. So it's actually probably add as well as peeling at the same time. But yeah, hopefully we were able to walk this through, yeah.
Kelly, I had just one question.
Yeah.
Can you say something about the relative affinity of RNA versus DNA to the H2A2B?
Oh, yeah, I didn't have a slide. So my postdoc did that Joseph. So he purified that part of the RNA, and it's in the order of 35 nanomolar KD. It's lower than DNA, but it binds. There's some specificity there. Yeah.
It's interesting to think about that mechanistically.
Yeah, it's quite puzzling, but to scratch our head, yeah.
One more question.
I think there's some recent data in yeast suggesting that different telomeres are regulated to have actually different lengths. I don't know if you would have any insight into this from your structural work, but do you have any thoughts on how that's being regulated?
I mean, telomeres with different lengths?
Yes, yeah,
Yeah,
at
it's
different chromosomes.
It's actually quite, it's a different kind of thing, but what has been shown, I think, by Joachim Linker is that it seems telomerase prefer to go to the short telomeres, because usually if you have telomerase in the cell, the telomere doesn't get longer all the time. It's actually stable. So there must be a way for it to maintain this stable telomere length. You know, too long telomeres is not good either. It's actually become fragile. So there's got to be a way for telomerase to know that probably prefer to add telomere DNA on the shorter one. So it's probably not coming to every telomere at every cell cycle. But how that happens is on its own, the whole field, yeah.
Great. Join me in thanking Kelly for a wonderful talk. All right, our third speaker, Elena Conti, is going to bring us full circle. Elena's career has really illuminated the important roles not only of RNPs, but also the importance of the fate of RNAs and understanding the fate of the transport and the fate of RNAs. And so today, she's going to tell us about that and some of the insights we can glean more generally about RNPs. So thank you so much.
It's okay? Yeah. So first, again, thank you all for putting together this amazing conference and also for the opportunity to discuss some of our work here. So our lab has been interested for many years on how RNAs are degraded. And today, Achim is going to present a poster on this. Achim is finishing his PhD, and he has managed to nail the cytoplasmic exosome, which we've been working in the lab for many, many years. He's going to present the poster today. So instead of talking about degradation, I'm going to talk about a different aspect we've been thinking about. It's just the other side of the coin, I think. You know, how are functional mRNAs protected from degradation? And of course, it's not mRNAs. We have to think about mRNPs. And the person really who has been the driving force in this is Fabien Bonneau in the lab. So we know about the architectural principles of many of these amazing RNPs. We have heard about it yesterday, this morning, and we're going to hear about in the afternoon. So what about messenger RNPs? And I think it's fair to say we expect them to be among the most diverse and dynamic RNPs. So what do we know? We know a lot about the part list. So we know the protein context. There are hundreds of mRNA binding proteins, work from Matthias Hense's lab, Roy Parker's lab, and others. We know mRNA has a tendency to fold. There is extensive secondary structure in cells, work from Howard Chang and Tom Cech and others. And work from many different labs over the years have looked at the biochemical and structural analysis of small elements in the messenger RNA bound to their proteins. And we can go from the 5', the open reading frame, many labs, many bits and pieces in 3' UTRs, poly A tail, and so on. So what about entire mRNPs? Now, some goes back to the early 90s, mid-90s, with the work of Danneholt on the Balbiani ring particles, and also more recent work from Roy Parker, Daniel St-Cluz, and also other work from Melissa. And Roy and Daniel, using single molecule fluorescence, have converged on this notion that mRNPs are more compacted when untranslated and more elongated when translated. And what I think I would love to do, can we take in some way the mechanistic understanding that we have here into the entire mRNPs? So this is what Fabien tried to do. So because we do not know the composition of individual mRNPs, and because we think the way they are the way they are built depends on the way they are made. So we have to take them from cells. And so Fabien set out to get nuclear mRNPs, which seemed a good place to start with, from budding yeast. And so he went on to try and isolate them with sufficient quality and quantity to study them. Now, of course, even if we can get a nuclear mRNP population that's very pure, it's going to be heterogeneous. Yeah, because we have all mRNAs there. So we have to adapt the structural approaches. We cannot use the same approaches we have been using for well-defined complexes. And what Fabien did is to harness two of the mRNA biogenesis factors that we know about, the Tau complex and SUB2. And these are really at the crossroad of transcription and export. Now, we worked on this complex, the Tau complex, a while ago. This is a dimer of 5 proteins, so it's 10 proteins, and this is in scale with the Pol2 and nucleosome, just to give you an idea of how big these things are. And this is just the core of the structured core of the complexes. So Tau is recruited co-transcription, and here you see all the groups that have worked on, not all the groups, some of the groups that have worked on this over the years. So Tau is recruited co-transcriptionally, of course it's evolutionarily conserved. It prevents the formation of R loops, and also mutants have mRNA export defects. And generally in the pathway, when you tamper with one of these mRNA biogenesis factors, you tend to have defects on all kinds of things. Now, Tau binds SUB2, which is shown here. It's a dead box protein, which has been involved in export. And in fact, SUB2 binds YRA1, which is another protein which has been involved in export. And mutants, and this we know from Torben Haik Jensen, Domenico Libri's work, mutants in these two proteins. HPR1 is one of the two complex subunits, and SUB2 ends up in what seem to be misprocessed in some way MRMPs that are targeted by the nuclear exosomes. So this seemed a good place where to start. And so Fabian set out biochemically to purify them, and as always, the devil is in the details. So I just tell you a little bit. It's a two-step affinity purification. We need 2 steps to remove the contaminants to get the purer. So normally, we get yeast strains, for example, so we start protein A, 3C cleavage, twin strep as second step. So if you have a normal purification, agarose beads, and you pull with one of the subunits of the TOE complex, you pull the other subunits. Now, the problem with agarose beads and with porous material is that you're essentially doing a size exclusion chromatography experiment. And so you're biasing for the smaller complexes, and the larger complexes are not retained. So if you get the same strain, and now you use non-porous magnetic beads, now the picture starts to be different. You get more material, you start to get YRA1, which is this mRNA binding proteins, and you start getting these other bands, and by sequencing, Fabian found, well, this looked like they were mRNA binding proteins. And so he reasoned that this, with a single bait protein, this was going to be too impure, because perhaps the TOE complex is not just bound in the context of the MRMPs, but perhaps it's in other places as well. And so instead of doing that, he puts the 2 tags on 2 different proteins. He protein A on one, twist strep on the other, and now the picture changes. And the pink, you get not only the TOE complex, but you see proteins like CBP80. You see NAP2 and PAB1. This has the poly-A binding protein in yeast. Of course, they are more abundant than CBP80 because they oligomerize. And you have this large amount of YRA1. We are not pulling on YRA1. We are pulling on HPR1 and SUB2. SUB2 is actually as abundant, probably, as YRA1, but we see an enrichment of this protein. And the other 2 things to think about is, of course, to be fast, because as soon as you open the cells, you have RNases and proteases, and Fabian can now it takes him 6 hours to go from the pellet to the cryo-EM grids, so it can be very fast. And in this way, he doesn't have to use cross-linking, so he can get these assemblies together in a native form. Now, this is, we are eluting in denaturing conditions, but of course we can also elute in native conditions. Pleural myostatis, Fabian has done this. And so this is now, it's eluting with biotin, similar purification, again, excess of YRA1, and now it's eluted in native conditions, so we can do a proteomic analysis and try and quantify the proteins by this intensity-based absolute quantification method. And you see here, YRA1 in large excess, you have NAT2, PAB1, and various proteins here. Enriched, but not so much. So what's YRA1? Now you see the number of people who worked on this protein. This is kind of an important protein. It's very abundant. It's essentially in yeast. It's non-shuttling. Expression is tightly controlled. Weird autoregulation. Splicing mechanism was studied by Chris Guthrie and also by Ed Hurt. It's recruited co-transcriptionally. Parclip data from the Kramer lab shows it covers the entire transcripts. Interestingly, if you look here from his paper, it covers, but then it kind of goes down when the poly-A binding protein goes up. So there is something there. And I love this paper by Gideon. And this was the person who really identified it as the major RNA annealing activity in yeast cells. So what does it look like? This is an AlphaFold prediction. It has an RRAM domain. It's a non-canonical RNA recognition motif because it doesn't bind RNA. As you'll see, it binds proteins. You have these IDRs, intrinsically disordered regions, which are highly positively charged, a couple of other helices, which are involved in binding SUB2. And it's concerned, this is actually where we know from work from Elisa, François Stutz, and others, that this is where the RNA binding activity is. And it's conserved in human. It's the AliRef protein. Now, this excess of YRA1 depends on the presence of RNA. If Fabian treats with RNase AT1 in the sample, what happens before elution? What happens is that it keeps the TOE complex. It keeps a little bit of YRA1. Actually, a lot of the RNA binding protein goes. CBP80 goes. The poly-A goes, and actually you see it here in the proteomic analysis. YRA1 goes down at a similar level at the TOE complex. You don't have the poly-A binding proteins anymore, and so on. So there's clearly RNA in the sample. So which kind of RNA it is? And so Fabian went ahead and extracted RNA, removed the ribosomal RNA from the samples and sequenced it. Now, he did a variety of analysis. I'm just going to show you in a couple of slides. So if we look at an enrichment analysis, what we have in the eluate versus the control versus the lysate, and we look at it and we plot against transcript lengths, what you see is that there is a positive correlation here. So we enrich in the purification via TO and sub2, we enrich in the purification for longer transcripts, which kind of makes sense because the longer the transcript, the more likely it's going to be that it has these biogenesis factors. And importantly for us also, because we are purifying, is that it's consistent with in vivo data. That sub2, WARE1, and this is again data from Patrick, are enriched on longer transcripts. So it looks like what we are purifying is biologically relevant. Now, the other thing I would like to show you is a couple of examples of where we're looking at the coverage of the representative transcripts. So what we see here, so here these are two biological replicates. You see the two lysates for a control. Here are the two eluates over here. And what you see is that there is a 5' bias, okay? There is less at the 3' end. So what we think is nascent transcripts that are not completed yet that we are pulling. And these are some examples. STRA1 is one of the largest gene in the transcripts in yeast, but it's more evident than in others. The other thing is that we have a few percentages, not so much, but of intron-containing transcripts. As you see here, they are enriched here. We have intron reads, and they are enriched in the eluates as compared to the lysate. And so what this seems to suggest, again, it's nascent transcripts, and what seems to suggest that at least the completion of splicing is not required to purify the particles and the samples we are purifying. So what do they look like? And the problem is, as I said, we don't want to cross-link, but it's, you cannot touch these things because they are not very happy. They disassemble. So Fabian looked at it. So he put carbon on supported grids by cryo-EM, and he sees this. He sees these shapes. They're all large, variable in size, and so clearly we cannot use the classical. You cannot do averaging on these samples. And so what Fabian did is to look by tomography. Now, I can look at this movie forever. But anyway, so here you see reconstructed. So he could get some specimens in free ice, so where you can see the particles in the tomograms. You see they're very condensed, different shapes. It's not a regular shape. If he extracts the particles, you see it here, kind of different, but compact, very condensed. So what compacts the nuclear MRMPs? Is there a kind of, I know it sounds weird, but is there a kind of nucleosome conceptual equivalent for condensing the messenger RNAs in the nucleus? And so to look at this, how do you look at these things structurally? So what Fabian looked is at cross-linking mass spec to look for proximity. Can we, we know what we have there. Can we see which proteins are contacting each other? So he cross-links with this BS3. And again, just as a caveat, we had to optimize the digestion protocols because these protocols for mass spec has been optimized for globular proteins. And as we all know, RNAs have unstructured regions with arginine and lysines. And so to be able to see these regions, you have to optimize the way you digest the sample. So there are intralinks, several interlinks. And so what Fabian then did here is to integrate the AlphaFold scores. These are residue by residue score, which correlate very well with how structured or unstructured the region is. If AlphaFold is very confident about how the residue is structured, then it's going to be structured. It's highly likely structured. So this gives you an idea. With the light are the unstructured regions of this protein, and the dark are the structured regions. And what we see here is that WIRE1 is really at the center of this interaction network. So we see contacts with the C-terminus of TO2, which is major components of the major scaffolding components of the TO complex. AlphaFold doesn't give a hit on how they two could interact, but we can zoom in on this interaction site between WIRE1 and NAP2, and by zooming in on where we see the cross-link, Fabian can use this and see, well, do we see something relevant by AlphaFold prediction? And he does. And now just to say, when, at least in the lab, we run AlphaFold predictions, what we like to do is to see how reproducible the predictions are. In 25 independent predictions, how many converge on the same structure. And so here, 20 more, over 25. The RRM actually at the backside seem to interact with NUP2. But the major cluster is interaction between YRA1 either with itself over here or with YHS7, which is an uncharacterized protein in the protein data bank. So I showed you this course. These are the cores that AlphaFold thinks these are, has very good predictions. They are very reproducible. But of course, can we put them, what do they look like in the context of the full-length proteins? And this is just to give you a flavor of what they would look like. So this is what we would look like if now on the core of the interaction, which maps the crosslinks, we put the rest of the protein by AlphaFold. So this is YRA1 with itself. Here are two RRM domains interacting with each other, and the IDRs are protruding with their positively charged and unstructured regions. Now let's look at YHS7 YRA1. Again, here we are mapping the full length on the cores. And again, similar thing. We have these long IDRs protruding away from the structured cores. Now, the tau complex, we don't see we can only say it's a proximity to YRA1. AlphaFold doesn't get a reliable contact. We don't know whether it's something it cannot model or whether, perhaps the interaction is mediated by RNA. But there are clearly, again, this IDR, positively charged and structured regions that we don't think about when, of course, I showed you the structure of the core. This is the cryo-EM structure of the core, but you have these long arms extending. So this is in a schematic mode what we think it's happening. And so we know from Gideon's work that YR1 has RNA annealing activity. So could it be that these other proteins that we see in the MRMP interaction networks have RNA annealing activity? And so we purified these proteins, of course, removing the nucleic acid components. Anybody who tried to purify these proteins knows the problem. And then Fabian ran an RNA annealing assay. So in this assay, he had, I don't know, I think it's about 300 nucleotides here. RNA, there is a complementary, perfectly complementary. So this is a proxy, for studying enhancement of RNA-RNA interaction, okay? Because never in a normal situation in an mRNA you will have this perfect complementarity. So for us, it's a robust biochemical assay. So there is a complementary part of about 60, and when you have annealing, you have this annealing part that will be resistant to RNase T1, which can digest, what is not double-stranded, cannot digest what's double-stranded. And we do the assay, try to do the assay in conditions that may reflect what we have in the cell, so kind of normal magnesium and so on concentration. So this is what it is when it's annealed at 65 degrees. If you do the assay 30 degrees, if we don't, these are the two proteins of the two RNAs. So what happens if you add YRA1 annealed, as Gideon showed? Now the RRM domain does not have RNA annealing properties, consistent with the fact that this is involved in protein-protein interactions, not in RNA binding interactions. Oops. YHS7 and the C-terminal domain of TOR2, this is just a C-terminal domain, the unstructured part, also have RNA annealing activity. Now, it's not indiscriminate, because if we take another component of the particle, sub2, which also has an unstructured region, but does not have these chemical features of being so positively charged, sub2 cannot anneal. Now, this is a slide, one of the slides I added yesterday evening, and this is because of the discussion we had yesterday. Now, there are small molecules, I think Phil asked about this, like spermidine. It looks like you have two kind of lysines, connected. So positively charged molecule. What happens if we put them in the annealing assay and if we pump up the concentration with this molecule? We do have some annealing. Yeah, it's not as good as YRA1 or the other proteins. So the other ones are really good at enhancing RNA-RNA interactions. And the way we think about them, it's a kind of flexible macromolecular counter-ion that helps bringing this RNA together. And I'm going to show you this from Jody Puglisi. It was shown again yesterday by Harry. there are not many examples of this in the literature. There are a couple of other examples, but this is the way we are thinking about on how these positively charged regions can come in and get this RNA together. And so this is the model that we have for this nuclear MRMP packaging in yeast, which is work in progress. So we think we have this structured node in the network. And from this node that you see here, there are these arms that extend and that are going to be probably in some dynamic way help bringing the RNA together. As I said, RNA annealing is a proxy. So what we imagine is happening that as the nascent chain comes, it's going to have a propensity to anneal, depending, of course, on how much complementarity there is in that area. Now, so this is in yeast. How about human? And it's, there is a very nice paper from Clemens Plaschka lab that came out very recently, where they purified with some differences, but a similar concept, human nuclear MRMPs via TOC1. It's the ortholog of HPR1. It's a bit different in treatment. They have nuclease and graphics treatment, so we cannot compare the details between the two because the preparations are different. But also what they see is that the particles, these nuclear MRMPs, are very compact. Now, fair to say they have a different model for how this mRNA MRMP compaction happens. That they propose is based on the oligomerization of the exon junction complex. This is based on a cryo-EM structure they have. But so what we ask is, well, is this IDR network model, would it be compatible with human MRMPs? And the orthologs are there. So what we did was to purify the human proteins and test with the human proteins, human AliRef, this is full-length AliRef, or the human C-terminus of TOC2, would they have the same properties? And yes, they have the same properties. The human proteins can also anneal, do have these RNA enhancing properties. And if we look at this, and I should say, AliRef is expected to be by itself, not in the EJC, it's expected to be in different complexes, whether you have intron-less mRNAs or intron-containing mRNAs in human cells. In intron-containing mRNAs, AliRef is expected to be as part of the exon junction complex or exon junction complex components. And if we now again use AlphaFold to predict the interaction, actually the interaction is quite similar to what Clemens is, but it can occur in a single EJC. You don't need the oligomeric form. And again, you have this module with these positively charged arms going out. Now, there is no clear ortholog of YHS7 in human cells, but data I have no time to show seem to suggest, I'll be happy to discuss it, that there is a conceptual ortholog in this complex of ERH and CH-top. And so what we think is that this model could be an interesting model to think about of how MRMPs are packaged in the nucleus. The beauty is that it can be valid from yeast to human, and also both for intron-less and intron-containing transcripts. And the question is why? The mRNAs can compact on its own. I mean, Dave Bartel has a very nice paper on this. So why would you need all these proteins? And it's interesting to think about whether perhaps, it allows a much higher order compaction, because we are a physiological magnesium concentration in the cell that you would be able to do to bring this, this negatively charged polymer in a very condensed form. And it could avoid collapsed or kinetically trapped states. RNA is very good at forming secondary structure. And it would offer also a way to do packaging in a way that's reversible and also regulatable, because as we go. I feel I'm the MRMP. As the MRMP goes to the cytoplasm, at some point, as it reaches the cytoplasmic side of the nucleopore complex, is gonna in some way unfold. And it's also regulatable, and I should say there are two papers on lysine ubiquitination of AliRef at these arginine regions, and also arginine methylation, lysine ubiquitination of yeast YRA1 and arginine methylation of human AliRef that are in the literature, where they show that this in special situation can help in the handover. And you could imagine this is something that you could disrupt and regulate the interaction. And with this, I would like to hand with 2 slides. One, to thank the people. This, the driving force has really been Fabien. And he was helped, Ingmar, Julia, Jerome, and Felix had input a different, for the purified proteins and various aspects, but Fabien was really a driving force. Achim has a poster on the exosome. We couldn't have done it without the facilities. I thought I would, mass spectrometry, sequencing, electron microscopy. Thank you for these funding agencies. And also thank you to the Vallée Foundation, because in 2019, just before COVID hit, we were here it allowed us to be here for a month. And this for me was a month where, you could just, stop and think and talk with people who know a lot about mRNPs and mRNAs and just think, what are, of course we were thinking about this problem, what is really the important questions that we could try and tackle in the next 10, 15 years. So thank you so much for the hospitality you've given us. And this is where we are. This is the present and the future for us. So thank you so much.
I have a question about if you were to induce ribosome collisions, would you end up with more of these compact mRNPs in the nucleus?
Sorry.
More or less?
Rewind.
If you induce ribosome collisions, do you end up with more or less of these in the nucleus?
I am not sure what, we haven't looked at that. But where are you, where are you heading at?
Well, is there feedback in terms
that
of...
goes back in.
Yeah.
This is brand new, so we haven't thought about it. So I'm really happy that we can discuss this. Yeah, haven't thought about that.
I had a couple of questions. I think, so the first one is, when you showed us the cryo-ET tomograms,
Yeah.
I wish you spent more time on it. You just
I can
concluded
watch it forever, yeah.
that they were compact, but do you see evidence for like RNA?
No, we are too low resolution, The problem is that it's, we are trying tricks to try and put this in, it doesn't like to go on ice. The particles don't like to go on ice, and we've been trying tricks, but every trick we try, the particles disassemble. So we have to try and get more of these samples and try to go to higher resolution or go to a different system. We don't see.
Okay, so I hope that works. The other question is, can you in vitro reconstitute particles that look like this from YRA1?
We have tried.
RNA.
As soon as you add RNA to YRA1, the whole thing aggregates in our hands. So we try and be clever. We increase the salt of YRA1. And of course, if you increase the salt, you add the RNA, it doesn't precipitate because it doesn't bind the RNA. So you decrease the RNA, so the RNA binds and the whole thing aggregates. So we haven't... Fabien dreamed the idea that perhaps one could add YRA1 in the transcription mixture, but it hasn't worked so far.
So, great talk. Have you been able to estimate the stoichiometry of the different components of THO and MEX67, YRA1 on either long or short transcripts by IBAC?
We haven't. We can try and estimate, roughly based on the COMASI, the difference in the COMASI. But also, this is a bit dangerous because, protein look very different. Different proteins look very different in COMASI. I should say that Ben Monpetit from UC Davis, using a completely different approach, is also coming to a similar, he's using fluorescence methods, and he's also coming to the conclusion that YRA1 is in abundance on the transcripts, and I think he has a much better method than we have to estimate how much YRA1 is there per transcript, and also, we have no way to check, depending on how the length of the RNA, but I think Ben has a better method to estimate that.
So could the extra layer of the EJC and higher eukaryotes be because their mRNAs are on average longer and you need greater compaction? Is that why you would need that layer?
So it could be. I think it's a caveat whether to me at least, it's still unclear whether the oligomerization, because the oligomerization was a truncation mutant of the EJC. So it's unclear whether in the context of the full length and of this domain that was truncated in cells, is it going to be the same oligomerization or a different oligomerization? So it's possible that it gives additional compaction. But the proteins I showed you at the end, which I just passed through, this ERH-CH top, this is a major hub, again, of proteins for compaction, which seems to be much stronger than the YH. It's a dimer that recruits another two factors. So I think in the human case, there is going to be more of this.
So, excuse me, over here.
Oh, sorry.
Great talk, interesting. You don't see HNRNPAs and Cs and the SR proteins. So are you stripping this in purification in some way to select for very stable complexes? Because exon junction complexes.
No, this was yeast.
Excuse me?
I showed you the yeast, the yeast
okay.
protein. I showed you the yeast protein. The human is not my work, so I don't know. I cannot answer. I mentioned the yeast. In the yeast, we do not see enrichment in splicing factors, but we don't know whether we just have few percentage of the introns are there. No, the work on the human has been done by Clemens Plaschka.
Elena, fantastic talk. So about, again, this paper that you cited, right, about with the human EJC, I remember in that paper, their model is that the RNA is kind of in the middle and the proteins are kind of like on the outside cradling and protecting the mRNP. And I noticed that in your model, you're drawing the proteins in the center and then like the RNA is like a cloud around the proteins. So is that is that based on data or is that like your current thinking or why this difference?
So as you notice, the TOE complex is outside in the model. We don't see the TOE complex in our data. I don't know whether it's a difference between species, whether it's, which I don't believe actually, whether it's inside the particles and we don't see it because of that, whether it's too small, yeast it's half the size of the human, we don't see it, or whether the nuclease, what they have is a nuclease-treated graphics cross-linked sample. Maybe, you maybe see something different in those conditions. So I think we need to look at this, using the same treatments, essentially. So if you do not use nuclease treatment and cross-linking, where is this? So it's too early to see.
Please join me in thanking Elena for a wonderful talk. And thank you, of course, to all of our speakers, and thank you also to our AV, because it's really nice to know that if anything glitches, it's covered.
We're going to accelerate the afternoon session a bit so that we can take the speakers back to their hotel before dinner. So please come back here at 10 minutes to 2 after lunch. We'll have a little shorter lunch break and then we'll start the session then. So first, just a word about Venki Ramakrishnan. He has COVID and he is stuck in his hotel room. He's feeling not real great, and also he's probably infectious. And he was incredibly disappointed to not be part of this event, not be able to give his talk. And he was feeling guilty about it and told me that he would pay for his plane fare. And I said, no way this could have happened to any of us, and we're sorry that it happened to you. But he was certainly an important part of organizing the program for this meeting. And so I do miss him. And he is such a terrific speaker as well. That means that we will have a for a shortened session this afternoon. The speakers are not allowed to ramble on endlessly just because we have one fewer speaker, because we're going to start the poster session early, and we are then going to have the speaker's photo at 4 o'clock. So all speakers and session chairs, please assemble out in the Itai sort of area there, and we'll probably go outside for the 4 o'clock photo. So the poster session will start at 3:30. The food will arrive at 4 o'clock, and the photographer will arrive at 4 o'clock. So it's my pleasure to introduce Aki Uhlenbeck, my good friend and longtime colleague. I was responsible for recruiting Aki from the University of Illinois, where he was very successful. He had a job offer at University of Chicago at the time to be department chair, and I said, Aki, come to Boulder. You can do science and you don't have to be department chair, and that was a very attractive offer. And at the time, he was well known for RNA-protein interactions, including the sort of iconic phage coat binding protein to RNA stem loop that we still use as a tool all the time. But when he got to Boulder, he decided to do something different, and over the summer he invented a hammerhead ribozyme transacting system and published a single-author Nature paper, and then really moved on to detailed studies of tRNA structure function in translation, among many other things. And Aki is also known internationally as sort of the glue that gets other people talking to each other in a very productive way. So Aki, why don't you start the session?
Thanks, Tom. I also feel terrible about Venki not being here. Let me just say one thing. Venki has written a book called Gene Machine, and I really recommend all of you read it. It's really beautiful in the sense that it's talks about its personal journey from undergraduate physics major in India to, big cheese professor at the MRC, and many steps in between. But it also tries to teach the reader about X-ray crystallography, what it's like doing science, what are things to avoid, and lots of other interesting stuff. So I really think you should read Venki's book. Okay, so I already noted several of the following three speakers. So all four of these speakers essentially have devoted their entire careers to the study of the translation process, or at least most of their careers. And so they have lots to say. And of course, all of them, knowing that Venki came out, have immediately started rapidly adding slides to their talks. So, just ignore Tom. We'll be glad to look at all your slides. So I love listening to all three of you. We're going to start off with Jody Puglisi, who is a kind of scientific sibling of mine because we both were educated in Nacho Tinoco's lab. First I as a postdoc, and later on Jody as a graduate student. He then really, at that place he was a co-discoverer of zRNA and started using NMR to study small RNA hairpins. He then went to Strasbourg and got introduced to tRNA and tRNA synthetases, moved to Santa Cruz, was a professor there very well, and of course got involved in thinking about ribosomes, and then not later moved to Stanford, abandoned his NMR machines, and started doing single molecule studies. And so today I expect, based on his title, that he is going to give us an overview of that process. So Jody.
Well,
getting set up. Thanks again for the organizers for this invitation to really what's RNA Woodstock to me. Just an amazing, and that's why I insisted that some of my trainees come to be at this event. What I'm going to tell you about is really the culmination of about a 20-year voyage where we tried to develop methods to kind of bring biological structures to life. I think, if anything, the talks of the last couple days, and especially this morning, have underscored just the amazing progress we've made in the ability to solve biomolecular structures of almost infinite complexity, capture those structures as snapshots in various and different states. But like a beautiful painting, those structures kind of stand there mutely, and really what we're trying to understand is mechanism. And if anything underscores the complexity of understanding a structure in terms of mechanism, it's that when a connoisseur such as Harry can stare at the ribosome and go, WTF, we still have a lot to learn. Second of all, what I'm really trying to do is animate these processes, put these snapshots together, and try to create a movie of biological processes in real time. And I'll show you our progress and the limitations of the things that we're doing. What we're going to focus on today was one of the reasons we started working on these types of approaches, is try to understand a really complex, although after splicing I feel like this is simple, process that involves the comings and goings of factors, messenger RNA, and ribosomal particles, which is the initiation of translation in eukaryotes. And that starts with an mRNA preparation period that involves recognition of the 7-methyl-G cap and a series of proteins, the AR4F factors. I'll talk a little bit about Carlos's poster about that. The ribosome lands. It's thought to land near the 5' end. And it was thought to use an energy-dependent process to kind of move throughout in the 5' to 3' direction towards the first start codon, okay, in a process known as scanning. There's recognition of the start codon, some more factors coming on and off, GTP hydrolysis, and then the joining of the other half of the ribosome, the 60S subunit. And obviously this is a very energy-intensive process. It's really the last step of gene expression, so it's highly regulated and has this complexity, like you saw in the spliceosome, of composition changing as a function of time. Okay. And this, of course, challenges lots of traditional methodologies, and unfortunately, Venki's not here to show you all the beautiful structural snapshots that he's been able to capture of various intermediates here, but you're almost limited to the stable intermediates that you can pull out and live long enough that you can actually study the process. And so this is challenged by unstable intermediates, this idea of compositional and conformational heterogeneity. The particles themselves are moving around, et cetera. And you know, this happens over a time scale that's not on the evolutionary time scale or origin of life time scale. We're kind of down to the more experimental time scales of laboratories. This happens on a seconds time scale in the cells, seconds to minutes time scale. And so we need methods that kind of embrace that complexity. And this is one of Venki's structures, and you know, I'm not going to do it justice, but it just shows how there's a 40S ribosomal subunit, the small subunit, RNA protein machine, and there are these enormous factors that help mold and guide the 40S subunit to do this particular function, which is to recognize a start codon. And so there's this large multi-protein complex that Jamie Cade has studied extensively that's very dynamic, that kind of wraps around the ribosome. And then there are these 3 proteins that are, that's a trimeric G protein that brings in the special tRNA, the initiator tRNA, and binds it into the P site of the ribosome. And there's a decoding process here that has to happen. There's start codon recognition. And you guys all learned the genetic code, the start codons in AUG. But there are near cognate start codons, CUG, UUG, that are also used. And so what is the basis of fidelity of start codon selection? Because obviously an error here of even one nucleotide leads to you making a protein that's of an entirely different sense. And just a little fun little tidbit here is the rules of start codon selection are kind of a mirror image of sense codons, in that the third position is conserved, what would be wobble in decoding, right? Whereas the first two positions, a little bit less conserved, and the first position can vary completely. So there's some interesting questions about start codon fidelity here. Once you kind of get to a start codon, right, you need to then make a decision, this is the right codon, and then all these factors which are sitting at the intersubunit interface. I'm looking down on the ribosome in this view. These factors need to depart because they act as steric blocks so that the other half of the ribosome can now join on the top, okay? And kind of a lunar landing to create all the RNA bridges that we've learned about over the years. Now, this has to process messenger RNAs in eukaryotes. These have 5' untranslated region, 3' untranslated regions. These are relatively short, about 100 nucleotides or so on average, a little bit less in yeast, a little bit longer in humans. Right? And then the 3' UTRs, of course, can be quite long in humans. And of course, RNA comes in all different shapes and sizes. I don't need to tell that to this audience. There can be long UTRs, short UTRs. There can be upstream near cognates. There can be upstream open reading frames. And obviously, ribosome, the messenger RNAs fold up into various structures. Great. Sorry. Oh, sorry. And just one feature here that is that the ribosome has to be essentially a helicase because the messenger RNA, if we do a cutaway here, in blue is the messenger RNA, and you can see there's a cleft where the messenger RNA sits on the small subunit, and really there's only enough space in most of that cleft for single-stranded RNA. And so there's lots of fundamental questions here about this process, even though it's been studied beautifully by biochemistry, genetics, et cetera, over the past decades, about many of the features about how does a ribosome get on a messenger RNA, how is a messenger RNA prepared, how is energy being used, how does the scanning process occur, how fast is the process, and how is the start codon recognized, et cetera. And what we're going to do is very simple-minded. It's essentially the molecular analog of real-time microscopy. We're going to stick fluorophores on all the various components, ribosomes, factors, tRNAs, messenger RNAs, et cetera, and we're going to track the process in real time. And we're going to do this at a single molecule level, quick single molecule primer. Okay, what we're going to do is immobilize messenger RNAs on a surface of various types. Right? And we're going to watch the comings and goings of factors as a function of time at a single messenger RNA level. That gets you the timing of the stoichiometry. When does factor 1 come on versus factor 2? The binding events are observed as bursts of fluorescence because you concentrate that fluorophore in an observation volume for a period of time. It gives off lots of photons. So that's going to give us time-resolved compositional or stoichiometric information. And then we're going to look at conformation in a very primitive manner. Right? We're going to use FRET. We're going to have a donor and acceptor dye, and they can couple to one another like little antenna. And when those two dyes come together in space, if you excite the green dye, it transfers energy non-radiatively to the red dye and gives off energy in the red. And that's just emphasized here. If I have an open conformation, you can see I have high green, low red. If I have a closed conformation, I have high red, low green. And many times the transition from one state to another is rapid. It's a single frame. That's because when you traverse a transition state, that happens rapidly. Most of the time in kinetics, you're waiting to overcome a barrier. And we can use this to look at conformation as a function of time. We look at lots of molecules, we do statistics, and you can build up kinetics by looking at statistics. You can also look at heterogeneous processes and kind of bin different pathways by which translation is occurring. The way I like to think about translation, it's almost like an estuary, or like the inverse of an estuary, where the rivers flow then inward and funnel the system in to a final event, which is 6S subunit joining, whereas the broad part of the estuary is the diversity of RNA structure and protein assembly. Okay, I always like to put slides like this in because we always serve these beautiful meals when we give talks, and we often don't talk about all the caveats and ugly things that are in the kitchen, okay? And one of the problems here with fluorescence and single molecule experiments is we have limited observables. I said I wanted to make a movie of molecules, but really what I'm looking at is a couple dyes attached to them. And so I don't have the degrees of freedom really to define things. And here's an example of a dance. I want to see a dancer, but depending on how I look at the dancer, okay, and shine light on the dancer, okay, I might get an entirely skewed view of what I'm looking at. I might think I'm looking at you know, whatever, flapping birds versus a dancer. Okay, so really you have to be aware of the limitations of the techniques that you're looking at. This is beautiful work that's been performed by a slew of great postdocs in the lab. Rosalind's here. You can meet them. Jinfan, Chris, and Mike are going off to set up their labs. And then you should go see the posters of all three of them that look at various aspects of this process. The early step of mRNA preparation is Carlos's work. Michael's looking at long-range interactions in messenger RNAs. We heard a little bit about that from Elena. And Rosalind's looking at the role of translation in disease. And I'm also fortunate that, you know, you get great collaborators. It makes you look better, right? And so I have great collaborators. Rachel's one of them. We worked on termination together. So the first step in any of these kind of processes, you got to roll up your sleeve and do good old-fashioned biochemistry. You've got to reconstitute a system. You've got to be able to put tags onto your system. You've got to prove that your fluorescently labeled molecule is still functional. There's just a lot of homework that you have to do before you can go off and do the glorious experiments. But let's say we did all that homework, and we did. That's why this took decades and not days. Let's do a simple experiment now. Let's watch initiation in real time. Right, so I'm going to start off with an unlabeled messenger RNA. In this case, I've pre-prepared it with the cap binding protein complexes. And I'm going to shine, you know, green light eye, and I'm going to detect green and red fluorescence, okay? And I have my 40S subunit labeled with a green dye and my 60S subunit labeled with a red dye in such a manner that when the two subunits are together, those dyes are close together in space and they can fret to one another. That's kind of a landmark event of initiation. So I wait for a while, okay? What I'm expecting to see is when the 40S subunit comes on, I should see a burst of green. When the 60S subunit comes on, green should go down, red should go up. That's an initiation event all the way to 60S subunit joining, okay? And then some photophysics occur that we can correct for. So now let's do the real experiment. We wait, we get a burst of fluorescence. We can do this on a large number of molecules. An mRNA that's been pre-prepared by this 4F complex binds a 40S subunit super fast. It's a very good substrate for the 40S subunit. If you don't do this, okay, you have to wait a long time. There's a long preparation period. Okay, you wait a little bit. So this is just showing you if you don't add the 4F complex, it just takes a long time for the 40S subunit to come on. And so we've done a lot of work on 40S loading. This is really a challenging subject matter, and we're just at the edge of trying to figure this out. Carlos's poster will be an example of that, of the RNA rearrangements that have to occur, the use of a lot of ATP energy to prepare that 5' end and kind of iron it out, as we heard about earlier. You know, we can talk more about that if you're interested. Okay, but now let's go on to translation initiation. So I get this burst of fluorescence, okay? And then at some later time, I'm probably scanning. Something's going on here, right? And then I get my subunit joining. And that time's about 20 seconds, which is about the physiological estimate of what an initiation time is. So we're looking at things that seem to be real here, okay? Which is always a fear. And you can see the fret from the subunit joining, okay? So let's now start to look at each of these steps individually. Try to understand what's rate limiting for that 20 seconds, because that's a pretty long time in biology. And what we're going to focus on here is that trimeric factor that I introduced to you, this EIF2. Okay, this is thought to be the gatekeeper of decision making. Am I at a correct start codon or not? If I'm at the correct start codon, I hydrolyze GTP, and this trimeric protein then disappears. It's protecting the 3' end of the tRNA. It exposes that 3' end for subsequent steps. Now, the GTP hydrolysis doesn't happen spontaneously. It happens stimulated by a gap, which is called EIF5. Okay? And there's some debate about whether it's stimulating GTP hydrolysis or PI release. That's not important here. Okay? And so this binds near the tRNA mRNA complex and another factor EIF1, right? And they binds in very close proximity to the initiator tRNA. And then subsequently allows the final factor, EIF5B, to bind to that naked 3' end of the tRNA and then chaperone the subunit joining process, okay? And the beauty here is those two molecules, tRNA and EIF5, are in close proximity. We can put tags on them, right? And we can start to measure things. And on the top is a turf microscopy view of just an equilibrium experiment. We're letting the factors kind of bind. We're not doing anything in real time. And what you can see is you see spots for the green tRNA here, which is immobilized on the surface, and you see spots here for FRET. And that's if I have a messenger RNA that has an AUG start codon. And you see nothing if you don't have a start codon. It shows you the specificity of binding of this factor. And you see this factor binds, the purple here goes up and the green goes down. You get very high FRET. When this factor binds, it's right next to the tRNA. It requires a start codon. Okay, so now let's do this in real time. You know, the system's working as expected. So we do the same experiment. In this case, we're monitoring the loading of the 40S subunit from fluorescence of the pre-bound tRNA. So that's a 40S loading event. Okay? There's no EIF5 present. That was thought to be a component of the 43S complex. It's not there. Okay? And then at a later time, you can see this burst of fluorescence of purple, okay, comes in for a few hundred milliseconds, and immediately afterwards, EIF5B binds. Okay? And that's just, you can do this statistically. You can show that when EIF5 binds, it's immediately followed by 5B. That tRNA is never there hanging out by itself. That ester linkage that has the methionine on it immediately gets bound by 5B once 5 gets on there and does its little GTPase activity. And most of the initiation events involve a single 5 binding event. Right? It just takes one binding event to hydrolyze the GTP. But sometimes the ribosomes get into a little trouble, and 5 fails, and you see these kind of futile events until finally on this final event, you get 5B binding, meaning I probably released that 2-factor and hydrolyzed the GTP. So, okay, that gets us to 5B. So what is 5B doing? Right? That's supposed to guide 6S subunit joining. So let's continue the movie, and you can see it about 10 seconds after 5B is occupying the 40S subunit. Now the 60S subunit comes on. I'm done initiating. So we can watch the whole process. We can do this with lots of factors, with labels to kind of map out the landscape of how this is occurring. Just one compact data slide, which is, okay, what happens if you start messing around with the start codon identity, okay, with the sequence context of the start codon, all the things we know from classical biochemistry. What happens is these late steps of initiation get more awkward. They take longer. It takes more time for EIF5 to bind. It binds less stably. It takes longer for EIF5B to bind. Okay? So you're extending the window of start codon selection when you start to mess around with the fidelity. And that's what all these different slides. So we're really starting to get at a molecular basis for how start codon selection is happening at these late steps of initiation. Now, what is EIF5B doing? I figured you guys saw enough structures this morning. I didn't want to show you any more. But this was a structure that, using the single molecule data as kind of a guidepost, a blueprint, a temporal blueprint, we can freeze samples at the right time, and we know the populations of everything. That's what we're doing here. We're watching an ensemble evolve in time. So we just freeze the samples after 15 seconds. We think we're going to have a population of intermediate that involves 5B bound. It's there. EIF5B is bound to the tRNA. This is the structure. Here's the tRNA. Here's this EIF5B. And here's a universally conserved factor, EIF1A. And the interesting thing is, what 5B is doing to gate subunit joining is exactly what I didn't think it was going to be doing. I thought it would chaperone RNA-RNA interactions between the subunits. No, the tRNA, if it's not positioned correctly, can't thread in to where it needs to bind in the peptidyl transferase center. So this is really acting like holding my hand when I try to thread a needle and get that tRNA in. So the tRNA is kind of an anti-association factor unless it's positioned correctly. So that's kind of fun. Okay, so where are we at now? Okay, we looked at all these late steps of initiation, but there's still 10 seconds of initiation. And is the ribosome kind of chugging along to the start codon slowly? Is it going to the start codon and ratcheting back and forth until it finally settles into the start codon? You know, how is energy being used, et cetera? So is scanning what's slow here? Okay, and so Jinfan in the lab, this is now shifting to yeast, but Carlos has done the same experiments in humans. What he did is engineer ribosomes where we have multiple labels, but in particular, we have a label at the prow of the ribosome. Okay, so as the ribosome will be scanning from the 5' to 3' direction, it should approach, okay, a probe that we're going to stick downstream. Okay, and this is just showing you this would be the 3' end of the mRNA comes out here, and we just put a probe on the ribosomal protein there using CRISPR technology or gene editing. And then we're going to put a molecule downstream of the start codon. We're going to watch the little choo-choo train go down the tracks. Obviously, this is on a flexible track, right? Single-stranded RNA, it's not a rigid polymer. You have to correct for all those things, okay? But what we're expecting to see is something like this. A ribosome should land at the 5' end if this whole model is correct, right? And then move directionally towards this static probe that we put onto the messenger RNA, right? And fret should go up as the ribosome moves. So this was a fun experiment. This is just showing you a two-color version of this experiment on an mRNA that has a 6D nucleotide UTR. And you can see just from this one trace, and the population behavior backs this all up, that the process is rapid. Red goes up rapidly, green goes down rapidly, and then there's photobleaching. It never seems like it goes backwards. It really is directional. You go from low red to high red, and you just stay there. So it doesn't look like the ribosome's ratcheting back and forth. It's really moving directionally. What happens when we make the UTR longer? Well, now you can actually see the steps. It looks exactly like what I did in my little diagram. You see this ramp. You see a ramp of fret. Green goes down as a ramp. Red goes up as a ramp because the movement is faster than the frame rate of my camera. So I'm not seeing individual states, I'm just seeing the directional movement. But the same thing holds here. It really does look like the ribosome lands at the 5' end, and we've done measurements to show this, and you move directionally, and you don't jitter back and forth. And the process is fast. Happens at about 100 nucleotides per second in humans, maybe, a little bit faster in yeast. Right? This is roughly the kind of timescales you get for motion of DNA binding proteins on DNA when people have measured this. It's fast. It's not rate limiting. I wanted for decades to study scanning. I mean, this is really why I started single molecule experiments. I'm going to figure out scanning. At the end of the day, it's kind of boring biology. It's fast. It's not where the action is. Okay, so just to end up here, we've been able to map a structural and dynamic landscape of how translation initiation is occurring. Loading requires reorganization of the mRNA. I didn't talk about that. You get rapid loading of the 43S complex that has the tRNA already bound to it. There's a factor called EIF1 that I didn't talk about that preserves the tRNA in what's called a P-out state that Venki was able to characterize. This allows the tRNA to scan along the messenger RNA bound to the ribosome, sampling rapidly, is that the right codon or not? When you get to the start codon, EIF1 departs. It really departs when you get there. It departs within a second or two. Okay? And then there's a period where EIF1 is sample, 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 and then EIF1 goes dark. And then at a later time, a decision's been made. I don't know what that is. That's our next step, is really to narrow in on what is the molecular basis of the decision making that allows the system to move forward into the final steps of initiation. So that's just a little view. There's lots of fun stories in the literature that we've published about what happens when you put mRNA structures into that. I welcome you to go read papers, but of course mRNA structure is an integral part of this process and can really modulate it quite well. At dinner last night, we had a little discussion. Why did you go do what you do? Like, who was the big influence in your life? Okay, I'm not stretching. The reason I'm here today is because of Tom. Okay, I was a terrible organic chemist. I literally blew up a laboratory. I didn't know what I wanted to do. I was studying chemistry at Johns Hopkins, and in my final year, I realized I really wanted to study spectroscopy and physical chemistry. And I took a class with David Draper, and I dug up my paper on enzymatic RNAs, 1984. I think I got an A, and that convinced me I wanted to work on RNA. So I went to graduate school in Berkeley, one of the few places where there was RNA research, the rest is history. I just wanted to say a few things more, and mainly to the students. Your scientific journey, it's like 40 years ago that I wrote that paper. Your scientific journey is long, okay? And, you're learning now. And I just think it's very hard to learn in the current landscape. There's so many methods. Science moves so fast. 1 minute you're CRISPR editing, the next you're doing high throughput sequencing. It's mind-bending. I'm a big fan of packing your suitcase for the long voyage with knowledge and properties and principles that are going to last you 40 years. Okay? Don't pack your suitcase with flimsy vests that they're going to disintegrate within 10 years. Learn something basic. Learn chemistry, learn physics, learn genetics. I don't care what it is, but a set of principles that will help guide you through that long voyage. The second part is the bridges. This was once I went to some retreat of Berkeley, and I went over the Golden Gate Bridge. And, when you're in the trenches of science, sometimes you get miserable. PIs, we go around, we grumpy. I didn't get my NIH grant. And you forget to look up. And, I've lived in California for 40 years. I used to live in New Jersey. It's miserable, And, you forget how beautiful the place is. And, I was crossing the Golden Gate Bridge, and you forget the beauty of the voyage you're on. So don't forget that piece, okay? Finally, this is the celestial bodies part. You go through this voyage, right, and you're like asteroids that encounter some very massive celestial body that sends you in a different direction, okay? And that's what is sitting here in this audience. It just freaked me out to realize people who have influenced me greatly in my life are sitting here. I'm going to go down the list. I started with Tom, okay? I would not be doing RNA biophysics if it wasn't for Aki. Nacho went to Aki's lab, learned how to do T7 transcription. I brought that back, started doing NMR spectroscopy. So thank you, Aki, okay? Next, I went to be a postdoc and then went off I was going to work on something really goofy in Santa Cruz. And then Harry sat me down, wrote a little diagram, said, Jody, you should work on this decoding site thing, right? So Harry taught me about the reason I work on ribosomes. Harry, it's you, okay? Thank you, Harry. All right, that's another one. Next, this is like kind of a negative thank you. Let's see. No, this is a positive thank you. I worked on decoding and really thought a lot about it because of Mike and the papers he wrote about decoding back in the day. And then people like Jamie, you, Jennifer, you guys, Venki, you started doing crystallography and made what I was doing absolutely useless. I was doing NMR spectroscopy of RNA, and I realized that was a losing game, and that was missing was dynamics, and that's what got me into this game. So thanks to all of you for all of that. Thanks again. Oh, sorry. This is the one person who should be here. This is Nacho. Okay, this is at my 50th birthday party. It's kind of happening. And these are some principles I kind of distilled of Nacho. That's for the students out there. Don't be wedded to your techniques. What's trendy and cool now, it's going to be pathetic looking 30 years from now. There's my NMR spectra. Have you seen an NMR spectrum at this talk, at this conference? No. So thanks. Thanks, guys.
Really nice talk. My question is, are you able to ever observe multiple 4DS joining events on your mRNAs? And is it related to the start codon or pausing the start codon or anything like that?
Yeah, so the question is, can you get multiple 40Ss on? Do you queue up 40Ss? We've done a little bit of that. It's editorial comment now. I hate supplemental material. it's like the bane of our existence. So Jin Fan did all these experiments in his Cell paper. Hidden in the supplemental material is that experiment. And it really looks like once a 40S is on, it's really hard kinetically to get a second 40S on. We need to do a little bit more to kind of map that out. But once you clear the start codon and probably release various factors, then you can load another one. So it doesn't look like there's 40S queuing. Yeah.
Jody, terrific talk. I'm curious about two things. So you get to the AUG and you hang out. But certainly in biology, sometimes you go past the AUG. Have you ever done that with poor codons and seen you go by?
Yeah. So in the yeast system, where we've done a lot of this, we put an upstream CUG. And what you see is about 10% of the time, you stop at the CUG. 90% of the time, you go through the CUG, you pause. You pause for about a second, where you're thinking, this might be, probably sampling some base pairing, but then you go onwards, okay? The interesting thing is, and this is the stuff I didn't talk about, if you start putting secondary structure around a start codon, the downstream start codon, okay, then what happens is during that waiting period, whenever you wait in biology, you can get into trouble, right? During that waiting period, the RNA that you melted, the hairpin that you melted, refolds on the ribosome, drives you backwards to the CUG, and you up the amount of CUG initiation. So yeah, we have looked at these kind of things, and we need to do a deeper dive, but that's a summary.
I've got the mic for one more question.
Yeah.
So I've always hated scanning. I still hate it, and don't have a clue why scanning evolved instead of just directly using the CAP and AUG recognition without scanning. Do you have a thought about that?
Yeah, I hate scanning too, so we're in the same boat. And there's a lot of features of scanning that are interesting. It really does look like scanning is not, I didn't talk about this, all the ATP requirements and the 4A requirements. Once a ribosome's on the messenger RNA, there's no external need of energy, okay? And so whatever 4A is used to load the ribosome, and we think that's at least a couple, that's the energy, the only energy you got plus binding that's used. And so, why scanning? I think it's for this kind of regulation of, if you're kind of landing as start codon and doing like you do in prokaryotes, you lose this ability to have a little bit more of a diverse menu of start sites like you do in eukaryotes. But that's my lame chemist answer to a biological question.
Yeah. Jody,
yeah.
Two questions. First of all, I've tried to do some of this kinds of experiments myself, and so most of you don't realize how hard it is to make all these substrates. It actually is hard. Yeah. Were all of that data that we looked at single molecule FRET, and those curves are summations of single molecules, or?
Yeah, so all the data, I'm sorry, I didn't talk at all about the methodology. All the data you're seeing are statistical analyses of single molecule behavior, right? And when I show you curves, that's the analysis of the single molecule. Of hundreds of molecules to 1,000 molecules, depending on the experiment. Yeah.
Well, the other point, the sort of, I don't know if you looked into this or not, but at least in the early stages of COVID RNA synthesis from the template, you have this initiation, and then there is this complicated phase change, codon shift. But it seems to me that this would be a technique that you could study that with. I would have.
It absolutely is. So I'm gonna admit a few things. One is we've been exceptionally remiss in taking this system and moving it to elongation. We did a great job with Rachel on termination. We did a great job with initiation. The signals to measure elongation in eukaryotes are just a little trickier. We did a lot of this in bacteria. We just don't quite have the ability to track elongation. We did putz around a little bit with SARS-CoV-2. It makes this protein NSP1, that binds to the ribosome and it blocks the mRNA cleft. And we did some single molecule experiments there that are kind of fun about blocking early steps of initiation.
One last question.
So are you also planning to look at the loading of phosphorylated EIF2 alpha because it reduces translation like heavily?
Where's Rosalind? Yeah, sorry, yeah. The answer is yes, of course, yeah.
Okay, thank you,
Thanks.
Jody. So, not long after I got to Boulder, in the late 80s, Jack Shostak invited me to Mass General to give a seminar about the hammerhead. And as often the case in these seminar visits, I was assigned to have lunch with the graduate students. This is very typical. And so when the time came, 3 graduate students came up and dragged me off, and I got hit with about an hour and a quarter of incredibly tense and difficult questions and completely drained me dry. So when I got back to Boulder a few days later, Tom asked, "How was it?" And I said, "Tom, we've really got to work on graduate student recruiting." I said, "No wonder Jack gets all this work done." The 3 graduate students that I had lunch with this day were John Warsh, Jennifer Doudna, and our next speaker, Rachel Green, who all ended up working on RNA in different ways. And Rachel also got inoculated by Harry in Santa Cruz, went to Johns Hopkins, and still works on translation.
Sort of.
Sort of.
Okay, thanks, Aki. That was really a lovely introduction. I want to thank the organizers for inviting me and giving me this opportunity. Yesterday was so much fun for me. It's like my entire training was in full display yesterday. Jerry Joyce and Jack and Harry, and it was a great meeting so far. What I'm going to tell you about today is recent work in our lab on how colliding ribosomes function as a signal for activating cellular stress response pathways. So I would say this is a little bit beyond the role of the ribosome in translation, and one might argue it was a pretty late evolutionary step. So I've gone a long way from group 1 introns. So I thought I'd start by sharing this. This is a cryo-EM tomography image from Liam Holt's lab of a yeast cell. So it's a slice of a yeast cell, and you can see in gray the organelles in the yeast cell. In yellow are some nanoparticles they put into the yeast to actually measure the viscosity of the cell. And colored in cyan are the ribosomes. And they can actually calculate the concentration of ribosomes in the cell from this at 23 micromolar, or 20% of the cellular volume. And in fact, in this study, they modulate the concentration of ribosomes by modulating the TOR pathway, and it changes the viscosity of the cell. So I think this is a pretty good way to think about how important it might be for the cell to monitor the activity of ribosomes in order to dictate what the cell should be doing next. So for a number of years, we and others have been interested in what happens to ribosomes when they elongate on messenger RNAs that have problems in the middle of them, problematic mRNAs. These problems can be encoded or they can be environmental. And the sort of things that we've focused on are the consequences of UV damage, or ROS, which can damage mRNAs and result in the pausing of ribosomes. They can't get past these damaged spots. And what we know is likely to be a problem is these incomplete nascent polypeptides are going to be toxic, and so the cell has evolved really great systems for clearing these species from the cell. And we refer to these as mRNA-specific quality control events. One of the things that happens is the nascent peptide gets targeted for decay. mRNA degradation is usually tightly coupled with this process because you don't want to keep translating messenger RNAs that are defective. And we actually began studying this with an interest in understanding how ribosomes got off these damaged messenger RNAs and back into the pool. And for many years, we and others wondered how it was that the cell could recognize a slow ribosome. There must be lots of ribosomes that are slow. How can you identify a slow ribosome? And in fact, in beautiful work from Hanny Zayer's lab at Washington University, he discovered that the key signature was not that a ribosome was slow, but that it was so slow that there were collisions that occurred. And what these collisions do is they present a new interface to the cell that allows for the recruitment of an E3 ligase. The E3 ligase ubiquitinates small subunit proteins, leading to the recruitment of this collection of factors, most of which have ubiquitin binding domains. So this is clearly what cells do to deal with the occasional problematic mRNA species to clear it from the cell. So you can imagine that this quality control machinery might be limited, however, and that's certainly the case. And what we discovered a number of years ago is that when the ribosome collisions exceed the capacity of this quality control machinery, what we found was that general stress response pathways were activated. These are referred to broadly as the ribotoxic stress response. It was deeply characterized by Bruce Magan from the University of Oregon for many years. And what we added to this system was really the idea that ribosome collisions were the key signature for those pathways as well. And so that's what I'm going to describe here. So a number of years ago, a great postdoc in the lab, Colin Wu, who's now at the NCI with his own lab, came up with the following set ideas about how ribosome collisions activate cellular stress response pathways. Colin found that ribosome collisions recruit at least 2 kinases in the cell. GCN2 is a well-known kinase. It stands for General Control Non-Derepressible, and it was discovered by Alan Hinnebusch studying what happens during yeast starvation. It's essential for, protecting yeast from starvation. And so that had long been implicated in ribosome functions, but not really in colliding ribosomes. Colin also identified the ZAC, which is a MAP3 kinase, which is at the top of a MAP kinase signaling pathway that binds to colliding ribosomes. Both of these factors, when they are recruited to colliding ribosomes, become phosphorylated, like most kinases do, to activate pathways. GCN2 has long been known to phosphorylate EIF2 alpha, which is a key factor involved in translation initiation. And when it's phosphorylated, it blocks ongoing initiation, preventing more ribosomes from loading on those problematic mRNAs. It also activates a transcriptional response, that integrated stress response for cell recovery. Downstream of ZAC, Colin showed, as had been previously shown actually, that downstream of ZAC are 2 p38 kinases, p38 itself, which is associated with cell cycle arrest, and JNK kinase, which is associated with apoptosis. And Colin reasoned at the time that the cell probably doesn't always turn on all of these pathways full bore, that the cell must somehow evaluate how many collisions are occurring in order to activate the correct cellular stress response. You don't want to fully kill all cells when you can actually deal with the collisions. And I will say that we were really largely unable to decipher that sort of level of regulation in this initial study. And so that's what I'm going to tell you about today, is our efforts to understand how a cell mounts a measured response to ribosomal collisions. This is the work predominantly of a postdoc in the lab, Niladri Sinha. You actually heard about his dicer structures from Brenda this morning. We've been collaborating with Alban Ortero at MSKCC for mass spec proteomics, with Sergi Rougeau, my colleague downstairs, who's a MAP kinase signaling expert, and Jeff Lee and Connor, 2 students Jeff in my group and Connor and Sergi's. I will say my 3 slides that I removed this morning were all of Sergi and Connor's work, so you can imagine it. It was live cell imaging. It was really cool, but it's gone. Sorry. Okay, so we got started by thinking about, does the cell really mount a measured response? And we just did a simple experiment with Sergi and Connor to simply ask what proportion of cells are either undergoing regular mitosis, these are, by the way, mammalian cells, or are undergoing G2 arrest, cell cycle arrest, or apoptosis, given varying exposures to UV irradiation, which causes mRNA damage. And you can immediately see that with increasing doses of UV, in fact, cells increasingly undergo apoptosis. So cells are integrating these responses. They don't treat all amounts of collisions as the same. So that was our clue to get started. What Niladri decided to do was to set up a large-scale proteomic experiment to decipher these pathways and their relative rates of activation. And to do that, he had to do a lot of preliminary biochemistry. And I'll just give you an overview here of the 2 methods that we traditionally use to trigger collisions in cells. This first one was developed by Hanny Zayer's lab, and the idea is you can take untreated cells where most ribosomes are not colliding. You can treat with high doses of elongation inhibitors, like anisomycin, and what happens is all the ribosomes bind the drug and are stalled. But if you use intermediate doses of drugs, the occasional ribosome binds, the lagging ones collide, and you can enrich for colliding ribosomes. So this is a convenient tool, but not very physiological. Most of what I'll show you today is the use of UV irradiation, which, as I alluded to, we've shown causes damage to messenger RNAs, pyrimidine dimers, that lead to ribosomes stopping and abundant collisions. So to get things set up, Naladri did an experiment where he took cells and he exposed them to UV irradiation and followed what happened over time. We can use general RNase digestion of cytoplasmic extract to produce monosomes in untreated cells, or some amount of colliding ribosomes in UV-treated cells, since at the collision interface, the mRNA is protected from digestion. I should say I'm not showing any structures, but there are beautiful structures of all of this, so they're real. What's shown here is the cytoplasmic digestion, and what you can see, these are monosomes, and these are the diasome species in untreated cells. We can blow that up, and then we can look over time. After 1 minute of dosing with UV, you can see a huge increase in the number of disomes and trisomes. And then you can look downstream at 5 minutes, 15 minutes, 30 minutes, 1 hour, 2 hours, 3 hours. And what you can see is that there's initially a really large peak of disomes and trisomes that are resistant to RNase digestion, and over time, the cell is resolving these collisions and fixing things. We can then look at pathway activation over the same time course, and what I'm showing here is the collection of westerns. Shown at the top here, we can focus on the ribotoxic stress response. We can first look at the activation of ZAC kinase through this phosphate gel, where the migration of ZAC is slowed down with multiple phosphorylation events. And you can see as early as 5 minutes, ZAC is really quite potently activated, and it peaks out as early as 30 minutes. We can also look at downstream substrates of ZAC, like P38 and JNK, and we see them strongly coming on in these early time points. For those of you who are concerned about DNA damage, many people are, you can look for the DNA damage response emerging. What you'll see is it emerges really quite late here in the 6-hour or 20-hour time point, suggesting to us that the primary response of cells to this UV irradiation is the ribotoxic stress response, and that the DNA damage response develops later. So with these data in mind, Naladri decided to look at a time course, this early time course when the ribotoxic stress response dominated. And he went on to do a mass spec experiment where he took epithelial cells, HACAT cells, which naturally respond to UV irradiation. He treated them with UV irradiation and then watched them recover over time, over a 30-minute time course. We did a phosphopeptide enrichment. We also looked at the total proteome. And you can look at the principal components analysis, and you can see that the time points cluster beautifully together, suggesting that the data are quite high quality. You can then look at those data, and this is what they look like. We get volcano plots. So on these volcano plots, in each case, we look at each dot here is a phosphopeptide that we can visualize. And in this one, we're looking at the 1-minute post-UV treatment collection of phosphopeptides. To the right are those that are enriched by UV irradiation. To the left, those that are depleted. And as early as a 1-minute time point, you can see the emergence of ZAC being phosphorylated, downstream substrate P38, and GCN2. They're already showing up as phosphorylated species, suggesting that we are truly looking at the ribotoxic stress response even at these earliest time points. We can look over time. This is the 1-minute time point, 5 minutes, 15 minutes, and 30, and you can see there's just a ginormous explosion of data. We can quantify 50,000 phosphopeptides here. And the question is, how do you go from this to thinking in any systematic sort of way about what's going on? Obviously, the cherry picking that I just demonstrated really isn't a great method, and it's cheating. So we've used a number of methods to try to sort out what's going on in these data sets. And I'm not going to tell you much about the clustering analysis, but you can sort your data into sort of the emergence of phosphopeptide species over time, and you can group them together and gain insight into which pathways come on at which time points. What I will tell you about is some of the insights that we've gained by using kinase inhibitors and knockout strains and deciphering those data using kinase signature assignments, which I'll describe. So one of the first things that we did to sort through these data, which again are overwhelming, was to use some inhibitors and knockout strains and repeat the phosphoproteomics to look at who was downstream of these upstream kinases. And this just gives you a sampling of those sorts of data. We have a knockout strain of ZAC, a cell line. We have a knockout line of GCN2. We also have beautiful ZAC inhibitors and GCN2 inhibitors, as well as P38 and JNK inhibitors, just thus hitting all of the nodes on this outline. And this is the data that emerges. You then redo the experiment, plus and minus UV treatment, and these are the sites that are sensitive to either the ZAC knockout or the inhibitor, and you would expect there to be overlap here. These are substrates downstream of P38 or JNK, and these are the substrates downstream of GCN2. And I'm just going to focus on one story from these inhibitor data to tell you some of the new insights we've gained. And I'm going to focus on GCN2. What happens to these cells when you put in GCN2 inhibitor or knock it out? So in order to decipher, so what you see here is that there's a lot of dots, and we'd like to know what's going on. Which ones go up, which ones go down, and who are they downstream of? One of the main tools we've used to decipher that, it came out of Lou Cantley's lab. What their group did is they purified 400 mammalian kinases. They then used arrays of peptide sequences that contained all the peptides of this length, and then they looked at the phosphorylation in the middle, and they just used p32 ATP, and they asked what is the specificity of each of the mammalian kinases. And that allows you to define a consensus sequence or motif, substrate motif, for each of these kinases. And those data were quite useful in allowing us to take our complicated volcano plots, such as this one for GCN2 in the presence of GCN2 inhibitor, and to identify what were the likely kinase-substrate relationships that were emerging here. So when we apply the Cantley analysis to this GCN2 data, this is what we see. We see that these are the main things that are upregulated in the presence of GCN2 inhibitor downstream of UV irradiation. And these are the things that are blocked from being phosphorylated. And I can tell you that over here there is a signature of VIF2 alpha and the anticipated substrates of GCN2. But what was most interesting was what emerged as being upregulated as a function of inhibiting GCN2. What you can see here is highlighted the most significant things that are upregulated downstream of GCN2 inhibition is actually the mTOR signaling pathway. So that suggested to us that in addition to GCN2 doing its known thing, which is to phosphorylate EIF2 alpha and to negatively regulate initiation, that another regular role of GCN2 when it's active is actually to limit mTOR. And mTOR is one of the main routes through which cells turn on translation of specific mRNAs and increase ribosome load. So it suggests that this well-known translational kinase is functioning both through this very well-known substrate as well as talking to the other major pathway that limits initiation in the cell. And that led to us to wonder that if we're blocking GCN2 function, what's then going to be happening is we're going to be abundantly loading ribosomes on messenger RNAs. We're no longer blocking ribosome loading. And so we wondered whether we would actually then hyperactivate ZAC, which is responding to collided ribosomes, to hyperactivate these downstream signaling pathways. And in fact, when you look at the other major thing that the Cantley analysis identified, it's the MAP caps here, which are known downstream substrates of P38. Consistent with the idea that one of the major roles of GCN2 is, in fact, to limit all sorts of initiation on mRNAs. And when you don't have that function, you're going to hyperactivate these downstream signaling pathways. And that has strong consequences for cellular fate. So I'll just show you a little bit of data to show you that we validated these predictions from the proteomic analysis. First thing I'm going to show you is what happens with UV treatment in wild-type and GCN2 knockout cells. You can see these are wild-type cells, and I've shown you this before. There's collided ribosomes in them that we can decipher. In a GCN2 knockout strain, we can see a strong enrichment of disomes and trisomes, consistent with the idea that what GCN2 is doing is preventing overloading of ribosomes. And when you knock out GCN2, you hyperload ribosomes on mRNAs. So that fulfills this one prediction of this major role for GCN2 in coordinating activities with ZAC. The other thing I'll show you is some Western blot data that also validates multiple observations. The first thing that you see here is on this Western blot is that in GCN2 knockout strains, there's a lot more ribosomal protein S10. In fact, there's not just more S10, there's more of all of the ribosomal proteins, consistent with the idea that one of the regular roles of GCN2 is to regulate the mTOR pathway, which was responsible for dictating the levels of ribosomes in cells. You can also see that with UV irradiation in the GCN2 knockout cells, you get hyperubiquitination of this small subunit protein, which is a key marker of collisions. So collisions are indeed increased. And in fact, we see hyperactivation of junk phosphorylation, which is this proapoptotic branch of the pathway, which is downstream of ZAC, again fulfilling the prediction from the proteomics data. This is where I would show you beautiful live cell imaging, and I'm not going to show it. But I'll simply tell you that the predicted consequence of junk activity being hyperactivated is that cells should be dying more readily. They should be hyperapoptotic, consistent with the idea that GCN2 is protective. And that's what these data show. We're now looking at cell death in response to UV treatment. This is the amount of cell death that's observed with this particular dose of UV in wild-type cells, and you can see the GCN2 knockout cells are really very sensitive to UV irradiation and really are on the path to death. And you can see that this apoptotic pathway is nicely blocked by ZAC inhibitor and by JNK inhibitor, as you would predict by this model that this is the route to apoptosis. So that's one set of information, or one new set of insights that we gained from our phosphoproteomics. What I'll tell you in the remaining few minutes is about another story, and it's more about ZAC, and it's really where we're going in terms of mechanism. And the question is, what can we get out of these sorts of data to understand something about this kinase ZAC and its function? Oh my gosh. Oh my gosh. Okay, let's try that again. All right. Oh my gosh. Okay, all my animation didn't work here. So let's start here. So this is the primary structure of ZAC. ZAC is a kinase, so it has a kinase domain at the N-terminus, a leucine zipper domain, a SAM domain, and then a largely unstructured C-terminal tail. And that C-terminal tail is known to dictate ribosome binding. If you delete that tail, ZAC doesn't bind to ribosomes and doesn't activate these pathways. You can look at the phosphoproteomics for ZAC alone, and there are 36 sites that are phosphorylated. And what you can do is you can use a clustering analysis to look at the patterns of phosphorylation. And you can see that the first cluster that's identified is here at the top. There's, let's say, 15 phosphorylation sites. And you can see they're distributed all along the length of the kinase. They're not abundantly phosphorylated, and it's pretty flat over time, so not particularly interesting, possibly. Cluster 2, however, comes up here, and these are phosphorylations that are quite abundant, and they rise and they peak around 15 minutes when we know P38 activity is maximal. And you can see that they're all clustered here right around the ribosome sensing domain, and so we wondered whether phosphorylation of those sites might regulate ribosome interactions. And finally, cluster 3 is shown here. It rises a bit more slowly and stays high, and they're scattered throughout. So what Naladri proposed to do is, instead of making single-point mutations, he decided to make cluster mutations. And so he took this cluster, and this cluster, and mutated all the serines and threonines to an alanine at the same time, and he looked for loss of function. So I didn't think it would work, just to be clear. It sounded like a long shot. He's very persistent. And in fact, the data were really quite promising. What we're looking at here is untreated cells, and we're looking at the activation of these pathways. Again, this is a phosphag gel first looking at ZAC activation. This is a wild-type cell line. This is a ZAC knockout line. We've put wild-type back in. Thank you. We've put in 2 kinase variants that are dead, either an active site mutant or an activation loop mutant. And then we either have cluster 1, 2, or 3 variants, or the all-cluster variant. And you can immediately see, even in untreated cells, that something interesting is going on with this cluster 2 mutant, which is that one near the ribosome binding region. You can see that there's some constitutive activation even in unactivated conditions. And if you look at the downstream signaling events, which is JNK activation, you can see it too is constitutively on even in untreated cells. When we look in treated cells, we don't see very much interesting. We actually see, mostly what we expect, which is the kinase deads are dead, and all of them get activated in ways we can't distinguish. But if I had those slides that I had eliminated, what you would see is that there's even some high activation here, and it stays on longer than it should. So we saw promising things, and it made us wonder if maybe the role of this cluster 2 collection of phosphorylations is something about turning the kinase off rather than turning the kinase on. It's staying on when it should be turned off. And what we realized, or we remembered, was that Simon Becker-Jensen had identified a motif right near the cluster 2 variants. That's actually a consensus motif for the Cullen ligase system, beta-TRCP, that when phosphorylated leads to ubiquitination and downstream degradation of the protein. So we wondered if that was the case. Naladri went on to do a mass spec experiment in conditions that were either inducing collisions or not. And he looked for proteins that associated with ZAC under collision-inducing conditions and proteins that were lost. With collisions, Naladri saw the enrichment of beta-TRCP2, which is the known adapter for this Cullen ligase system. Naladri saw that ribosomal proteins were depleted from ZAC, suggesting that ZAC might be dissociating from ribosomes as a consequence of collisions. So that led to a model where ZAC recognizes colliding ribosomes, becomes phosphorylated on cluster 2, leading to ribosome dissociation, and ultimately targeting by this beta-TRCP Cullen ligase system for degradation to regulate pathway activation. And I can tell you that both of those predictions turned out. What's shown here is a normal polysome profile in untreated cells with either a wild-type ZAC, kinase dead, or a cluster 2 variant. And you can see what I haven't shown you yet, which is this beautiful association of ZAC with elongating ribosomes. ZAC is always associated with elongating ribosomes. But when we expose cells to UV irradiation, you can see that wild-type ZAC starts to fall off and is found at the top of the gradient. Kinase-dead ZAC doesn't fall off, and our cluster 2 variant doesn't fall off. So if you can't phosphorylate cluster 2, you don't dissociate from ribosomes, and you don't fully activate, you can't go on to next steps. The final piece of data I'll show you is that there's degradation downstream of collisions of ZAC. And you can see that here. This is a Western blot simply looking downstream after cells are treated with collision-inducing anisomycin, and ZAC is degraded, as you can see quantified here. Kinase-dead ZAC is not degraded. Clusters 1 and 3 variants are fully degraded, and the cluster 2 and the all-cluster variants fail to be degraded, consistent with the idea that this phosphodegron is critical for downregulating ZAC post-collision. And finally, the final prediction of ZAC not being turned off and not being degraded would be that you would be pro-apoptotic. Cells that can't be phosphorylated on these positions should also die more readily. And indeed, that's what we see. We see that the rates of apoptosis are increased for the cluster 2 mutant and are very low for a kinase-dead variant. So those are the data I want to share, and I'll just close with a final model. I hope I've convinced you that we've discovered some new property for ribosomes. It's not exactly an RNA-driven event, but I would say ribosomes are mostly RNA. So at the center of this cellular stress response pathway is that the cell is detecting colliding ribosomes in order to activate these key signaling pathways to lead to an appropriate cell fate response. We've added this new layer where GCN2 doesn't just regulate the phosphorylation of this well-known initiation factor, but this new regulation through the mTOR signaling pathway in this coordinate response. And that feeds into regulation of ZAC because that regulates ribosome load and the activation of this pathway. And finally, I've told you about this new back regulation, or what's it called, autoregulation of ZAC, where ZAC regulates its own activity downstream of collisions through degradation, presumably allowing the cell not to overactivate. It needs to recover from stress before it starts activating again. And finally, in closing, I would say that one of the things that we're thinking hard about is, I set this up by showing you this view of a cell and suggesting that ribosome activity should be an important thing that the cell pays attention to. But you can imagine that the balance of whether the cell can fix the collisions or undergoes apoptosis might be very different in different cell types. And in neurons, there's very little translation happening. We would expect very few collisions, and yet toxic proteins should be very problematic. And so maybe these pathways are most strongly activated. Whereas in other cells, maybe pancreatic tissue that's pumping out proteins, you don't want to be responding to every little collision because there's too much load. And so we're thinking about those problems and how the cell balances these decisions in different cell types. And with that, I'll close and thank again Naladri, who really led this entire project, Jeff Lee in my lab, Alban at Sloan Kettering, Sergi downstairs and his student Connor. Jake Saba I brought, he has a poster here. He's actually looking at TOR in a different context and top mRNAs, and I hope you'll get to his poster. And Luke Hantley's lab for helping us to analyze the data, and to my wonderful lab. Thank you very much.
Amazing. What do you think the bumping event is?
The what?
The bumping event.
Oh my God, I can't
One ribosome bumps into another. What do you think?
What do I think happens?
Yeah.
Well, I think there's some data to suggest that they can frame shift. It can lead to frame shifting in certain systems. So if you don't kind of control that collision, it can lead to frame shifting. I think the main thing it does is it recruits stuff. I mean, I think you get some interface that is a composite interface, and there's not enough binding energy unless there's two ribosomes next to one another. But I will say, we don't have great examples. So the E3, nobody's been able to capture, and that's the one you'd really want. We, with Roland's lab, have captured an endonuclease that sits between ribosomes, and it binds to both sides. It binds to both of them. We've also captured this protein called MBF1, which was Naladri's first project, where it's a collision sensor, and it's right at the interface. if somebody asks me about ZAC, I'll tell you I actually don't think it's going to recognize an interface. But I think it recognizes two close ribosomes. So, Brenda.
So you sort of have a correlation with inhibiting GCN2 or other conditions that you see trisomes and tetrasomes. And do you think that's just a correlation? Do you think that those are necessary? For example, if you looked at trisomes, would you see less ZAC? So are the signals that increase?
Do you need, is the higher order structure necessary? Yeah, we don't have, I don't have anything strong to say. I will say Toshi, who works with Roland, thinks a trisome is the best substrate.
Right, but you might say some of the changes you're seeing might be more concentrated on the trisomes if they're really directly connected.
Yeah, they could be, but we certainly don't make that claim. We certainly just have a correlation.
So in the degree of apoptosis induction here seems impressive. How many, are there chemotherapeutic compounds who hit translation without hitting DNA? And have they ever been explored as therapeutic agents?
That is a great question. There are. So one of these new PROTACs targets ERF3 or 1, I don't remember which one, which is a termination factor. And certain cells are sensitive to that drug and others are not. I would predict it would cause collisions at all the stop codons in cells, and I would predict it would hyperactivate these pathways, and I don't know that they're thinking about them. But I think yes. I'll just also admit that it's a sledgehammer that we used here. We used a sledgehammer of UV irradiation, and if we were to do it again, I would back off. But
last question?
Yeah, I was going to ask Rachel about the. So you showed really nice data on the timing of these events relative to DNA damage, for example. But obviously there's, with UV, for example, there's DNA damage, there's message damage, there's transcriptional effects, all of those things. So but your event seems to be quite early, so I'm wondering how you think about those responses being integrated in terms of the ultimate cellular response. Right. So in terms of the physiology, how do you think? Is your thought that this is early and therefore this is causative, or this is early and therefore this is somehow integrated with other types of damage to give you an ultimate cellular signal? And if so, how are you thinking about that working in terms of physiology rather than
rather than the sledgehammer? Yeah. I want to go on the record as saying I'm not denying DNA damage is fully going to be on here and totally relevant, and they certainly are going to be integrated. I think our evidence suggests that the earliest response, and certainly by MAP kinase, is the ribotoxic stress response. And our best evidence was in our first paper. We damaged an mRNA in vitro and transfected it into cells, and that fully activated these pathways. But if you look in our volcanoes, check one is up, things are happening. So I think it's how the cell integrates. I think a lot of the DNA damage responses wait for replication forks to, for them to coordinate S phase, et cetera. So we would have a lot to do to really say we were only studying this. Thank you.
Our last speaker of the day is John Atkins. Perhaps due to his contrary personality, he studies the parts of the translation machinery where things that go weird. You get frame shifting, miscoding, hopping, mistakes in initiation. I don't know why he does that shifting. Yep, must be. So John has spent his whole life, he spent a lot of time in Utah, but he's a professor, has been for a long time in Cork. And I think we're going to get several examples of weird stuff.
Yeah, okay, thank you. Okay, so yesterday Tom introduced us to the RNA Tag Club and to GAMOF, and I want to continue with the culmination of this work to understand the general nature of genetic decoding was the Crick et al. Paper in 1961. And this is a caricature in three-letter human words of what they did. So Crick started with a putative frameshift mutant in phage T4 and isolated revertance of it. And from the almost wild-type revertance, he separated the compensatory mutant that was present and then showed it had characteristics of frameshift mutants, and then isolated several more from that successively and combined them in various ways to deduce that translation starts from a fixed point and is read in non-overlapping sequential triplet codons, and critically, that there are no commas involved. No one told the COVID causative virus that there were no commas, and it requires a single frameshift during its decoding to make its replicative proteins. And important for this is a comma, which is a three-stemmed pseudoknot, in which the stem 1 and stem 2 coaxially stack, and stem 3 is perpendicular to it. And it lodges at the entrance to the mRNA channel and blocks between the head and the body of the ribosome and prevents the translocation. And this structure, which was obtained in Nenad Band's lab, was at sufficient resolution to be able to see all the ribosomal RNA modifications, most of the protein modifications, and it shows that the loop, a flipped out base in the loop 1 of the pseudoknot was interacted with the amino terminal domain of S3, which the carboxy terminal, which was known to be part of the unwinding complex of the helicase, but it hadn't been known about that part of that protein. And it also revealed the entire path of the nascent peptide exit tunnel, and there was a specific interaction there, and both mutants of that and indeed of the base in the loop caused a great reduction in the frame shifting. And there were numerous other features revealed in this structure, which I won't go into because it's published, but I'll be happy to talk to people afterwards about them, how this influences the thing. What I do want to go on to say is that several groups had worked on a compound called MTDB, which was claimed to inhibit this frame shifting. And in our hands, it did not do so. But Jungji Guo in Yale his group published in bioRxives a paper that a compound called merifloxacin did inhibit the frame shifting. And we tested that and found that it did in a growth-dependent manner, concentration-dependent manner. And actually, it inhibited SARS-CoV-2 infection of tissue culture cells, and then a half maximal concentration was 4.3 micromolar. So it's not in the nanomolar range, so it's a proof of principle that you can get inhibition of the frame shifting, which is required for the virus, and it inhibits the viral propagation. Now, in human decoding, there are only 2 frame shifts, -1 frame shifts known, and both are retrotransposon arrived. But this compound had no effect on the cells. They were very, very healthy. So it's only a proof of principle, but nevertheless illustrates a different approach that could be used. And unfortunately, my then graduate student and postdoc, Pramod Ban, he had to cancel from coming here 3 weeks ago. So how did we get involved in this? Well, the answer was a long time ago. So I was intrigued by the Crick et al. Paper, which I heard about 2 weeks after it was published, in early 1962. And I decided to switch to science and graduate in genetics. and because it raised so many questions in my mind. And then I went on to do a PhD, not that I wanted to register because there was no jobs, but I did anyhow because I was interested in following up on this. And my supervisor, after a while, went to Australia for 6 months, and when he came back, he had lost interest in science. So I and another graduate student were basically free to do anything we wanted on a side topic. And so there was a paper just then published from a well-known American lab characterizing putative frameshift mutants and showing that they were only considered to be frameshift if they were non-leaky and they had no external suppressors, because no one at the time knew that decoding of Titan required faithful translation of over 30,000 codons. But the presumption was that frameshifting would be so deleterious that selection would have developed multiple ways to prevent this from happening, even to the point that you couldn't get a single mutant of a translation component which would compensate even locally or cause it even to happen even locally. So what we did was we took a putative frameshift mutant in the tryptophan operon of Salmonella and placed it on minimum median to get revertins. And we characterized just looked on day one, we saw the big ones, which we guessed were site revertins, and the semi-fast, which are probably analogous to Crick's compensatory mutants. And indeed, they mapped within the gene of interest. And many of the slower growing ones, the laggards, mapped in a separate gene externally. But we also concentrated on minute ones. We were interested in those very much and characterized rather a large number of those. And one of them turned out to be in the same gene here. And when we separated it, we found that it was a base substitution mutant as judged by mutagenesis and not a frameshift mutant. I'll come back to that one in a minute. So I just then want to go in, then cloning and sequencing were developed. And so the early 80s, we got to go back and sequence our frameshift mutant. And it turned out that we had, the original one was a frameshift. It lacked an A. And one of our mutants was of a valent tRNA, so it read from the GUG to the overlapping GUG. But actually, one of our mutants, a rather weak one, turned out to have two ones. Clearly, there was an initial mutant of what we now know to be a release factor 2, yes. And it was overtaken by a mutant of L9, which we, many years later, found restrained forward mRNA slippage. And we characterized a number of mutants of the tRNA, which I won't go into, but they were in different parts of the body of the tRNA. One was even of the first substitution of the first C of the CCA at the 3' end. And here I just want to show one of them on this decoding seat here. You shift from GGA back to GGG. This is a mutant of tRNA GLI2. And so this was done in 1983 here. Where is this decoding seat? It is in a sculpture. So a friend and I commissioned a sculpture actually for the 60th anniversary of the double helix in the National Botanic Gardens in Dublin. And so the decoding seat is here on a mound, and it looks across at the DNA, which is a classic Charles Jenks type structure, which is in smaller ones are in Clare College, Cambridge, and in Cold Spring Harbor Lab. And so there's a connection between the DNA and the RNA and the DNA. The RNA doesn't completely control the DNA, even at this stage. And there's been discussion at this meeting as to where life originated. Well, I can tell you where it originated. It originated there. And the evolutionary trajectory, there was advances and then some partial fall as it rises the moon, so it rises the mound. So there's an underlying upward trend, but it wasn't universally upwards. And of course, we had to have the key molecule represented here on the upward trajectory. So I want to thank Tom for the model of the structure on which the aluminium structure represented here was based. but more importantly, I want to thank Tom for the magnificent role he played in the RNA World books and in the associated publications in Colping Harbor Perspectives in Biology. And I want to thank both Toms. I'm thanking you. I want to thank both Toms for this great meeting. So thank you very much, Tom. So sorry, I need to go back 1 second. Yes, so the minute mutant, was a change of a C to an A. And so we had an AAG decoding tRNA, which we guessed read back, and it went to AAA. So you could have a base substitution mutant, which partially compensated for the frame shifting. Apologies. Okay, so however, that was in 1983, so I didn't want to wait that long. There was a publication from Princeton in 1970 where a person had characterized a number of frameshift mutants, and they dismissed all mutants which were leaky because they couldn't be frameshift. So that provided a real opening to me. So I wrote and got all the genuine frameshifts, the non-leaky ones, and with a more sensitive assay, showed that they're all leaky. And then teamed up with Guarini to show that indeed, with his fidelity mutants, that there were leakiness because of a translation level phenomena. And so, if anyone who gets cold sores or shingles, they're often treated with acyclovir. And Don Cohen's lab have shown that some of the herpes mutants which resist acyclovir, have a frameshift mutant near a shift-prone sequence. And so a small amount of the product can reactivate the virus from latency, but not trigger the inhibition by acyclovir. Now, there are quite a large number of pseudogenes in the known, but some of them make some product. So they're really pseudogenes because of the leakiness of the indels that they contain. Now, in various cancers, it was initially, shown in melanoma, that the interferon gamma that produced leads, by indel 2,3-dioxygenase, to a tryptophan deficiency. There's a kynurenine pathway effect. And Rudy Agami's lab has shown recently, with a number of publications in Nature, that with the tryptophan starvation, there is some frame shifting, which leads to peptides, aberrant peptides from the new frame. And also, there's mischarging of the tryptophan tRNA then, in the absence of tryptophan, with phenylalanine, and this gives peptides as well. And these are an immune display, and this is a possible entry point for intervention for immunotherapy. But the sloppiness involved also helps the cancer with stress. So for the cancer, it creates liability, but it also gets over some problems. So these are non-programmed frame shifting, but the most extreme example that we know of is in ciliate euplotes, where Eigner and Czech and Klobuchar and Farber and others found that some frameshift mutants are very leaky, extremely leaky, and there are quite a number of genes that actually have frameshift. So, one example with Lobanov here, what we did was we did some ribosome profiling. And I should say that in this organism, that for termination is when you have UAA or UAG very close to the 3' end, as if this poly-A binding protein has to interact with the release factor to allow termination. So they're recoded, if you like, to specify termination. But in fact, when they occur within coding sequence, they specify frame shifting. And so the most commonly found one is with AAA. You go to AAU in the following sequence. So you have a UAA stop codon, but in fact you get very efficient frame shifting, basically 100%, or else you can go -2 from AUA to AUA, overlapping way. And in this paper, we found that there are a large number of P-site codons, but they're in at a much lower level, so it's not equal depending on what happens, and we don't necessarily know that it's all repairing, by the way. And also you can have UAG in the A site instead of UAA. By the way, tryptophan, UGA is just reassigned to tryptophan codon. So then the next thing, which was a very recent paper, which is actually going to be published this week, is we have sequenced the transcriptomes from 8 divergent eupleusis species and identified where is an insertion and is it more frequent than the absence of a base, depending on the phylogenetic thing as to where it occurs within the phylogenetic tree. And so the gains actually exceed losses by 10-fold, or approximately, but the potential sites where you can have changes to make that is about 30. So basically, we think it's totally neutral or approaching neutrality, so that there's effectively no selection. And so the drift is actually irreversibly committed now to getting an increased number of frame shifting sites. And so we estimate that depending on the particular species eupleuses, that they're going to end up between 17,000 and 71,000 frame shifts without being programmed. Okay, so I want to turn back to the original caricature, and this will show why I chose these particular words. So if you take this R here and you frame shift to REC, you get the word recode, yeah, and you get recoding here. Okay, so together with Ray Jesland, we developed this word to indicate dynamic recoding. And so at the translational level, although as I mentioned, there are counterpart effects at the transcriptional level. So there are temporary changes in the rules, in the decoding rules, and it's the competition with standard decoding. So a proportion of the product is of a new type, or else it can be used for regulatory purposes. It is a recoding site at which it occurs, and it can be affected by stimulatory elements. And there are several types, one of which is a dynamic redefinition of codon meaning, and that is completely different from context-independent codon reassignment. The same word has more recently been used by Jason Chin and George Church and others for genomic purposes, where it also makes a lot of sense. It's just to make clear that there are two different things that are being discussed in here, and I think it's valid for both of them. So our first recoding signal that we found was associated with the release factor 2 autoregulatory frame shifting, which was found by Katsky. And so what we have here is we have a Shine-Dalgano sequence, just 3 nucleotides before the shift site, which is too close, and it tends to shove the frame shifting forward. It's quite a good stimulator, actually. And it took some years before we noticed that actually in DNX frame shifting, which is -1, that there's also a Shine-Dalgano stimulator. But it is 10 nucleotides, 5' of the frame shift site. And it stimulates the -1 frame shifting. And then by moving the Shine-Dalgano different distances further 5', we're able to deduce then that the pairing happens and the ribosome can continue to move. And it's approximately 14 nucleotides from the break point where the elastic band breaks, if you like, before this happens. Now, so we always think of the Shine-Dalgano as being used for initiation, but the evidence from this, in fact, is that as ribosome is translating, it also scans mRNA. The anti-Shine-Dalgano sequence scans mRNA for potential complementarity. And so then we wondered if indeed that might have played a role in primordial ribosomes to sort of tether the message to the RNA to prevent diffusion in a protoribosome. And Shine-Dalgano pairing, by the way, the Shine-Dalganos would be a glycine codon, which is possibly an early amino acid as well. So Shandalganos are in archaea and in bacteria, and probably really ancient. So take that for what it is. We don't know, obviously, but we're just wondering about that. So then come to the next point. So if you have something like Gag and Paul briefly overlapping, and there's a long Paul here, then there's obviously a problem with how is Paul expressed, and it could have been either transcription or translation, but it was, as we well know now, that there's ribosomal frame shifting here. But what happens if you have just a frame shift, an internal position, make a short thing? That's much less obvious. So I had lucky to get a postdoc who had done his PhD in astrophysics in Cambridge, and he came to us and he developed a program for looking at looking at synonymous codons and then summing them depending on whether there was 1, 2, 3, 4, or 6-fold redundancy, and based on the difference from a neutral evolution model, and then statistically bringing them together and summing them and averaging it over a sliding window. And in arterioviruses, he found an example here of an overlapping gene, which I will come to. It was actually the second frameshift site which was known in arterioviruses. But he also found a number of candidates then, which we went on to show with collaborators in flaviviruses. And the distance between the stimulatory structure and the shift site is a common 5 to 9 nucleotides in eukaryotic cases, that is. But in the arteriovirus, the distance was much longer spacing. And in fact, it was just a linear sequence. And it turns out that there was a protein that bound to this. And so presumably, it was then further removed from the frameshift site when it had its effect. And one of the other cases that was found in this study was in cardioviruses, when EMC, which had been a modeled message in the early stages of eukaryotic translation, never had this hidden thing. So in a way, we call these cases of where you have hidden things, it's the equivalent to genetic steganography. So if you have a code with the cryptography, when it's obvious there's a code, but if you read a message and there's a hidden message in there, that's steganography. So this is genetic steganography, if you like. And Andrew Firth, together with colleagues, he's moved back and he's got a position now as a biologist in Cambridge, and they followed up on the study of the proteins binding here and this thing over here. Now, I want to go on to the next case, who was actually in flu virus, and I discovered an overlapping sequence here with the protein PAX, which plays a role in viral shutoff of host gene expression by cleaving the host mRNAs. And this was miserable in terms of frame shifting because the efficiency was really low, but it does illustrate the range of different products you can get from this. And we collaborated with people who showed the importance of this, and the main work actually was involved in them. However, the shift site was unusual. This was a +1 frame shift, by the way. And then taking that shift site and then looking for sequences within the human genome, we found the ASXL one is a good candidate for utilization of that. And in fact, we since have evidence that this is a real case. Now, Andrew went on to find many more cases, which because of what Thomas said, we certainly won't go into, but it affects replicase, capsid protein extension, accessory proteins, but also it led to the identification of many cases of leaky scanning, which hadn't been appreciated. Now, I mentioned that there's a transcription level counterpart of this, and one of the cases where in bacteria, for instance, if you have a homopolymeric run, of greater than 9, or so greater than the normal hybrid length, you can get overlapping and you can get a series of extra bases inserted. And so there's a counterpart of DNX ribosomal frame shifting where there's transcription slight. And we also studied a case with the insertion sequences where the nascent RNA forms a structure within the RNA polymerase, and there's one base missing, as it were, in the product. However, the one I will illustrate is in the potiviruses. Now, potiviruses, the potivirus family, constitutes about 30% of all plant viruses. But there's about 50% of the economic loss due to plant viruses and potiviruses. And here, it was thought to just make one polyprotein, but in fact, it makes an overlapping one because of transcription slippage at G and 6As. G and 5As, at which you get an extra A when it slips. And the extra product is required, actually, for viral movement. Now, one of the most extraordinary, the boom-burst cycle that was first known about, at least in the Western world, was in the Netherlands in the 1630s, where there was a mania for tulips of a particular pattern. And the most prized one got actually outrageous prices, and it was this one represented here. But the people who bought it weren't able to breed true from it. And the reason was because it was actually due to a potivirus infection. And this pattern is due to the movement protein, which is coming over from this way over here. So, okay. So, and I'll switch to a cellular gene which uses frame shifting in a regulatory sense. And this is a gene called antizyme, which is a negative regulator of polyamine synthesis. And the gene for antizyme is in two parts. It's the main part is down here, which is in the +1 frame, and there's a UGA at the end here. And this was, so when there's low polyamines, there's more termination here, and there's less antizyme made, and this means less inhibitor, and so increasing polyamine levels. On the other hand, when there are high polyamine levels, there's more frame shifting, making more antizyme, and this leads to decreasing levels. Antizyme acts by inhibiting the synthesis of putrescine and through ornithine carboxylase, which leads to spermidine and spermine. But it also affects the uptake, the permease. But in addition to that, has cross-pathway effects. And there are actually 3 different antizymes. Antizyme 1, the first one was found, and antizyme 2 is different, and it has an association with MYC, as Murai and Matsufuji have shown more recently. And then antizyme 3 is in developing male germ cells, and it's the only one expressed there, whereas antizyme 1 and 2 are in all other tissues. So we've been studying this. Now, it has a pseudoknot, but remarkably, the pseudoknot is only 3 bases from the shift site. And so it should be unwound, in fact, by the time of the action of the frame shifting. But we've shown that, well, there's a PGP sequence up here, which is generally probably recognized by EF5A, or facilitated by EF5A. And it's actually in the stop-go 2A phenomenon as well. You get the same sequence, in fact. And so there's a synergistic effect between the pseudoknot and translation through here by mutants here. But there's also other effects of the pseudoknot just in this level. And there's a tryptophan codon here, and there's a complex mixture of things here which are relevant to this frame shifting. And then together with Ninad, we've done some cryo-EM on this, and there's a spermidine in the at the position where the hypozine of EIF5A would be present in the ribosome. But so there's much to sort out here about exactly how this thing works. It's really quite a challenge, and we need Jodie's help for that. so then I'll come to read-through, where you get a C-terminal extension, so it can be regulatory, and one of the cases is in the plant ERF1. This is not our work, where there's an effect of the read-through by NMD and the exon junction complex, and it was done by other people in 1997. So what we've been studying initially was the taking tobacco mosaic virus, which was found in Cambridge to have a read-through in the early 70s. But we looked at the context, and it was CARYYA was the important feature for TMV read-through. And it turns out this is important for a very large number of plant viruses which use read-through. In contrast, in human, we have only a modest number, not more than 12 or so, of human read-through causes, and in some of them the context is CUAG. In some others, there are elaborate structures, and notably in one of them, for which there's the strongest sort of bioinformatic evidence for read-through, we can't get it to work at all. So there's something, I'll come back to that in a minute. But in other cases, we found that we had experimental evidence for a pseudonaut in murine leukemia virus. So it's the counterpart of MMTV, if you like, the GAG-POL. But the pseudonaut in this case is relevant to stop codon read-through. But Alan Miller has found with beet western yellow virus that the component, part of the component in the stimulator is 4KB3'. And so this may be related to the trying to prevent the logjam of translation going 5' to 3' and replication going the other way. It may be a component of that. More recent work together with Lynn Cooley's lab in Yale. A long time ago, Ian Cooley noticed a case of read-through where the extended extension after read-through is enormous and actually greater than the normal termination product. But together with her, we had a Western expressed in different tissues, and there's really quite a remarkable specificity of the read-through in the central nervous system. And this is most markedly in the adults. And so there's the mechanism by which there's such tissue specificity for read-through is unknown at this stage. But in Drosophila, the number of read-through cases is approximately 800 of that sort of order. So it's really quite widespread and very, very different from the situation in humans. Now, the 21st directly encoded amino acid, we call it, is selenocysteine, there are 25 human genes, 25 human selenoproteins, and nearly all, there's a single UGA which specifies selenocysteine. And Mari Berry, a long time ago, found that the stimulatory structure is a structure called CSIS in the 3' UTR. And more recently, the structure of this has been discovered in Berlin in this particular case. We've been more interested in the protein, the one that has multiple UGAs. This is selenoprotein P, which is a carrier protein taking selenium from the liver and various tissues. And it has two CSIS elements. And the second CSIS element is primarily involved at the first UGA, and the first CSIS is primarily involved at the other UGAs. And the efficiency here is not very high, and the efficiency here is high. And we've been studying making mouse mutants of the mouse gene of the stimulatory elements involved, and studying it in that way, and comparing it to what other Marley Berry in particular had done in Xenopus, to see in the endogenous situation the effect of it. And we don't know in detail how the CSIS talks to the oncoming ribosome, and there's a pass-off between one of the CSIS and the others. But in addition to that, we've done some bioinformatics and noticed that in oysters there are 46 UGAs, and there we have fed oysters selenium and done ribosome profiling, and you can make full-length product. So it's effectively unitary efficiency of specifying a selenocysteine in the latter part of the thing. And our maximum actually, and there's an enormous accumulation of selenium in the tissues in these things, and this may be due to heavy metals or some other thing like this. Our maximum is freshwater mussel, where we have 132 GAs or 131 in the 3 part of the coding sequence. The first UGA is involved in redox, I might point out. But however, other organisms, like a spider, has just 9, and in aquatic organisms, there's a much greater level of selenium. Okay, finishing up shortly. Okay, just one more. Is this right? Talk about this? Just bypassing 50. No? Okay, all right. So I'll skip the gene 60 bypassing, talk to anyone who wants about it afterwards. So I'll just summarize that we have been studying frame shifting, and this is a motif from the book of Kells here. The head's motif to indicate directionality. The messenger RNA is at this side of the ribosome at the beginning. It's gone through the ribosome at the end. The nascent peptide is here, and at the end it's out. And there are 3 different types here, frame shifting, and some bypassing down here. And then I just want to thank the people involved. And so Ray Jesseland is in gold here. He's actually a fabulous collaborator for many years, and I really appreciate his help in Cork. The key people have been Pasha Baranov, Andrew Firth, and Gary Loughran. And Pasha Baranov has his own lab, it's overlapping mine, and Andrew is back in Cambridge. Who else? Avala Ivanov was 20 years with us and is now in Tom Dever's lab. And external collaborators, there are of course many. I want to thank obviously Jody very much for the Gene 60 stuff, which he did critical work, which I wasn't able to talk about, and Marina Rodnida also in that, and of course Ninid Ban, and Martin Ryan was in the stop-go, the 2A phenomena, which I won't talk about, but yeah, okay, thank you very much.
I think we have to go to posters. I think we'll have to skip questions because we're running pretty late. So thank you, everybody.
Okay.